Hello, everyone. On behalf of the organizing committee, I would like to welcome you all to the day 5 of our program, First Human Cell Atlas Latin America Single Cell RNA-seq Data Analysis Workshop. We all hope you've been enjoying our program. We are at the last day of the lectures for the first week, and I hope you're able to appreciate the diversity of the topics. I would like to thank the organizing committee and the scientific committee for this meeting. As a reminder, the organizing committee and scientific committee included people from Mexico, Chile, and Brazil as a first joint force to train people in single cell analysis in Latin America. We partnered with people from the Human Cell Atlas. This is the second event that targets Latin America, but we have colleagues attending this workshop from different parts of the world, including the US, Europe, and Asia. During the second week, we have a hands-on workshop with trainers based in Brazil, Chile, and Mexico. This training part of the program is also virtually, but the trainers are located in these 3 countries. So we have welcomed people from other places of Latin America, including Argentina and Colombia. We would like to thank specifically Christine, Tracy, Luke, Samantha, and John from the Human Cell Atlas, who helped us a lot during the organization of this event, and the institutions that supported us in different ways. Our program today looks like this. So 2 lectures now in the morning, followed by a 45-minute break, and 2 lectures in the afternoon, another break, and the project design discussion with people who have been selected for the second week. We would like always to remind you to check out your time zone so you are in time for the lectures, and that during the breaks you're supposed to log off and then come back in at the time of the meeting. So welcome you all. I'm going to now introduce our first speaker of the day, Dr. Jay Shin. So welcome, Jay. In 2008, Dr. Jay, hold on a second. So Dr. Jay Shin acquired his PhD at the ETH Zurich, Switzerland, after his research training at Harvard Medical School in Boston, USA. During this period, Dr. Shin combined both experimental and computational approaches to build gene regulatory networks involved the angiogenesis of the skin inflammation. Dr. Shin then joined the Riken Institute in Japan after receiving a special postdoctoral research fellow, where he established single-cell genomics technologies and analytical tools to investigate the role of stem cell differentiation and reprogramming. Now, as a team leader, the lab collaborates with world-leading industries and research institutes, and currently co-leads FANTOM and Human Cell Atlas to comprehensively characterize all functional genes and cell types in the human body. Welcome, Jay.
Can you all hear me?
Oh, yeah.
Great. Okay, then I will get started. Thank you, Patricia, for your kind invitation and your kind introduction. Hello, everyone. My name is Jay Shin, and I am a team leader at Riken Institute in Japan. And today I will give you a broad introduction to single-cell data analysis, but mainly how we can start to explore different types of single-cell modalities or single-cell technologies to investigate how genes are regulated and how epigenome is modulated and so on. So I'd like to start with this map here on the right-hand side of my title slide. I really like this picture because this is, of course, an outline of a human body, but it's also a map which shows you a traffic, but also a roadmap to where cells can be, a particular point can be connected to another point of the human body. And as some of you who have driven a car or have been in a bicycle, you have to follow traffic laws, and there are regulations and rules that tells you which way you have to go in order to make sure that the traffic does not get into an accident. So just like in our human body, all of our cells have regulations and traffic controls that allows us to function and allows the cells to function properly so that our body can maintain its healthy state and homeostasis. And of course, a disruption in any of these regulations can lead to a severe congestion in traffic sense, but in a human sense, it can lead to disease. So not only profiling these single cells are important, but also understanding how genes within these cells are regulated is critical to understanding the human cell atlas. So I believe you guys have seen these slides, this slide many times, but I just want to emphasize this rapid development of single cell technology over the last 10 years or so. And now it's really very much feasible to profile up to hundreds of thousands, even millions of single cells in a given experiment at a reasonable cost. And this rapid development has allowed us to profile not only easily accessible tissues, but very rare tissues, and to discover very unique cell types in the human body. But not only the platform, but the workflow that allows us to profile these single cells have become very much streamlined. And this is really an effort of global community coming together to provide the right understanding of how to dissociate cells from a tissue, to how to prepare our certain cells in order that we can, accurately profile the cells and be able to sequence them in order to acquire the right information that we need. So I think we should really leverage this opportunity to take advantage of all these streamlined processes that have been developed and try to generate the data as quickly as possible. And I think this is really the right time to expand and contribute to this global efforts to single cell data production. And combination of rapid advancements of single cell technology and this streamlined process of single cell preparation, there has been a tremendous and groundbreaking reports where people were able to profile hundreds, thousands, or even hundreds of thousands of single cells of the human body. And this is a report published by Guoji Gou's lab in China last year, where they profiled millions of single cells and identified various tissue types of the adults, as well as the fetal developmental human body. And similarly, Jay Shandor's lab in University of Washington also have profiled tremendous, over millions of single cells of the fetal development. Here you can see the diversity and the heterogeneity of the cell types from various tissue organs. Similarly, the human cell atlas effort have allowed us to profile multiple tissues, but also allowed us to profile from various individuals to define really what is a meaning of healthy liver or healthy kidney. Because if you just profile a liver from one individual, you can understand their general gene expression pattern, but it is very difficult to say that individual liver sample was a healthy reference. So really, to truly build a healthy reference that we can all rely on, we need to generate these single cell data from multiple individuals as much as we can in order to understand where the variations are and where the minimal variations are found. And where that minimal variations are found could potentially be defined as a normal or healthy reference. And that really is going to be a very difficult task, but again, a very important one in order that we can understand the health state of the human body. You know, even though the atlas is still very much in the process of developing, in the recent years has been extremely useful in understanding the role of the COVID-19. And in fact, ACE2 protein and TMPRSS2 protein are receptors that is recognizing the COVID-19 spike proteins, proteins. And you can see from the human cell atlas data, the granularity of various cell types found in various tissue organs that express these two receptors, such as ACE and TMPRSS. And you can see that in nasal cavity of the human nose that the ciliated cells and secretory cells express these two receptors, which makes a lot of sense because ciliated cells will then attract the virus, and then that's how it gets entered into our system, as well as the lung and the type 1 and type 2 alveolus expressed as well. So again, the power of the human cell atlas can really help us to define potential entry points of various virus types out there, and hopefully this can be used for future therapeutics and preventions as well. I just want to highlight that in order to build a human cell atlas, we also have to take into consideration tremendous amounts of technology standardization. Because we want all of our data to be reproducible, it's comprehensive and predictive. And there's, within the human cell atlas, we have a technology standardization working group. And this is a report that we have recently written to summarize our efforts within the working group and how we can collect various samples from various countries and various labs, and even profile in different technologies, and then try to better understand how we can integrate various platforms so that we can, again, define the healthy state of the human body. So the working group aims to provide standardized operating protocols to its members, to all of you, in order to generate data that can be minimizing the batch effects and really enhancing the biological effects as much as possible. And hopefully we'll continue to provide toolkits and suggestions of how to keep track of metrics early on, in order that later these metadata can be then further utilized for batch correction. So I know that the lectures this week are all about data analysis, but I just want to emphasize that data collection and standardizing the data collection in order that you have a high quality data will only help you with a downstream data analysis. And if you really have a poor quality data, I think you will be pulling your hair out all the time trying to analyze this data and try to make sense of it. So I would highly encourage you to really start on the right foot by ensuring that you have highly standardized data production pipeline. I also think that this is going to be much more complex when we look at sampling procedures, because if you look at individual variations, as I mentioned earlier, the genetic variations and sex variations, the age variations can also contribute to a large number of heterogeneity in your data. So having a very accurate recording of your information is helpful. Even genotyping their DNA could also be useful. Also, depending on which part of the organ that you're sampling can determine also types of cells to expect and the quality of samples that you'll get. So recording where these tissues come from are also critical in order that all the data that we generate can then be integrated in the future. So I just want to, again, emphasize, collect as much metadata as possible in order that the data can be used for not only for batch correction, but also data integration with all the other community data sets. So in Asia, we are attempting to start these data collection efforts. And although immune cells are relatively easy, since we're just drawing the blood from individuals, we have encountered so many challenges of protocols of how we purify the cells, to what kind of technology versions we should use, which analytical tools. So even the simplest samples that we can collect, we already encountered a lot of hurdles. And you know, what kind of questions we should ask the volunteers, the donors. What are the exclusion criteria in order that we can ensure that we have only the healthy individuals. And these are, you know, not trivial, and I highly encourage if you are interested in building an atlas within Latin America, just starting with the PBMCs, blood samples, and getting the discussion going will really be helpful. And again, this is started out with Japan weekend and Korea in Samsung Medical Center by Unyoung Park and Shayan Prabhakar, whom gave a lecture earlier this week from Singapore. And recently we have new members from Thailand, India, Russia, and Saudi Arabia. And again, the goal is to collect over 170 individuals from each site. In total, we'll have 500 from 3 countries, but in addition of these 4 countries, we hope to collect over 1,000 donors in order to understand how the different variations of the human genetics contribute to the gene expression patterns. Okay, so this is called expression quantitative trait locus analysis, or EQTL, but it may sound very complicated, but simply what it is that we try to understand the genotype that are different across the individuals and ask whether that genotype has high expression or low expression. And by plotting this box plot here from GG genotype to GT genotype to TT genotype, that we can measure the expression is actually down or negatively correlated. So we can say that this genotype leads to low expression of a gene, and therefore it may potentially lead to a particular phenotype in the cell or in the body. So all of this is a huge effort also outside of Asia, and there's even, in fact, a single cell EQTL consortium, which is a much more global effort. And we hope to, hopefully one day we will be able to work with Latin America to get a population cohort that we can generate and compare data with Asia, US, Europe, and rest of the world. So if you guys have any questions about how to start this up, please reach out to me or Shayan Prabhakar from Singapore. And it'll be really great to standardize our methods in order that we can integrate our data together. And this is all possible because wow, 150 individuals, that's so much data and probably very expensive. It is expensive. At the same time, we are trying to minimize cost by the strategy called pooling. So you can pool, at the moment, 116 individuals into a single, here, 10x genomics run. And what that allows us to do is we can sequence their transcripts. And because the RNA can also contain the genotype information, we can use this genotype information to demultiplex the 16 individuals into 16 data points here. So here is a UMAP depiction of the human PBMCs from a single 10x reaction, but then we can deconvolute them into 16 different data points representing for each donor. And we can tell because here we have 2 males in the 16, and these 2 males are expressing the Y chromosome gene called RPS4Y1. And here we can clearly identify 2 males, and rest of them are females. We also use what's called batch donor control. So these, every 16 has 1 donor control, which is always same in every 10x run. And this donor control is done in Japan, in Asia, in Singapore, in Korea, always the same donor. So we're now using this information to batch correct across various countries and various experiments. So to make the long story short, has been a lot of effort. It's still very much ongoing, and we are now able to integrate data not only from Japan, but Korea and Singapore to make this very comprehensive gene expression profile along with the genotype information. So that was just an example of what we are doing, and to also emphasize the importance of collaborating across different countries and to do large projects. But at the same time, you know, single cell analysis does not have to be so big. It can be very focused and biological driven experiments. So here is just a few consideration. Again, is, you know, if you have a question that you want to address, some of the things that you have to consider is how big should the sample size be? What kind of technologies I should apply? How many cell numbers do I need to profile? What kind of analysis should I perform? And all of this sampling technology, annotation, and analysis workflow needs to be really well standardized, and across the collaborators who may provide samples and collaborator B who may provide the technology needs to be well communicated. And I just really want to emphasize how this whole workflow needs to be standardized from the beginning in order to ensure that you can interpret your results and validate and publish. But of course, before you begin any of this exercise, asking the right question and the right hypothesis is critical. And hopefully I will touch upon this question a little bit more in the future slides. So I think the often questions that I get is, well, what can you do once you have single cell RNA-seq data? What kind of things are possible, and what kind of things can we learn from single cell data? So I thought I'd just summarize some of those things, kind of the rudimentary things that we can gain from single cell analysis, such as, okay, we can maybe define a new cell type or a new state that transitions between the cell types. Or we can just identify key biomarkers and gene signatures, or just perform differential gene expression between 2 cell types or 2 conditions, or accessible regions, chromatin accessible regions. We just want to compare maybe healthy versus the disease, treatment versus the control, or understand their pathways. Are they involved in cell cycle pathways? Are they involved in metabolism pathways? So these are, I would say, kind of the discovery phase, where with a single cell analysis, you can relatively with ease, you can probably generate a lot of these information, as long as, again, your data is well standardized. Now, probably the next component is a little bit more challenging, but probably really worth exploring with the single cell data, which are, we can reconstruct transcription factor regulatory networks, or maybe signaling pathways using some of the antibodies that target signaling molecules. Or maybe you can understand cis-regulation. So what I mean by that is maybe chromatin interactions like high C, or DNA methylation that regulates the promoter accessibility, or simply enhancers or promoter activities could be considered cis-regulation. Or we just want to understand modules or cell lineage or cell-to-cell communication. And I think you guys have already seen some examples by experts in these individual topics, how to do this. And I hope that more and more single-cell data can become possible to explore and elucidate out of these regulatory processes. And then the third thing that single cell is possible and equally contributing to the discovery and regulatory is the genetics component, where variants, variants or copy number variation or structural variations or somatic mutations at the single cell level can definitely influence what kind of genes are expressed and what kind of regulatory network it defines. So understanding these genetic components in the context of gene expression is critical, as well as population genetics. As I mentioned, the eQTL example, or pharmacogenomics, how different drugs can respond differently to different individuals because of the genetic background. So what I want to emphasize here is that while we can do single-cell RNA sequencing to understand the cell types and genes that are differentially expressed, with a genetic component, we can understand even much more accurately why these genes are differentially expressed or chromatin accessible regions are different. So things that we have to consider is then, should we perform multiple single-cell modalities, meaning should we do single-cell RNA sequencing and single-cell taq-seq and single-cell genomic sequencing, or is there a way to do single multi-modalities? So is there a way to do single-cell taq-seq, RNA-seq on the same single cell? So I'll try to touch upon some of these examples in the next few slides. So here is an example of why it would be so important to have a copy number profiling, because this is a SKBR3 cell line, which is highly ploidic, and you have about 83 chromosomes, I think, in this cell line. So there's a large number of copy number variation. And as you can see on this panel here, you have this genomic DNA in the diploid here with 2, but you also see a lot of variations with the high numbers of genomic DNA, and that correlates very well with the gene expression. Here you see in the mean expression. So you can see that more copy number you have higher gene expression. So just simply, if you look at gene expression alone, you think, oh, they are upregulated or downregulated because their promoter was activated. But no, in fact, it was upregulated, or it was highly detected because there are a higher number of genomic copy number. So understanding this is very critical, especially in cancer and cancer studies, obviously, because you have many copy number variations in that context. So please keep that in mind and think of how you can address some of these issues going forward. So I just want to highlight reasons for performing multi-omics, as I mentioned, is cancer, because in copy number variations and somatic mutations can easily influence gene expression. But there are other examples where in development, where you have very rapid cell state transitions, chromatin accessibility can be not synchronized with the gene expression. So if you do, say, chromatin accessibility assay like a taq-seq on one cell and then RNA expression on the other cell and try to integrate these two data sets, you may not actually get a very accurate dynamics because the transitions are happening so quickly where chromatin changes may precede gene expression. So again, another reason why you should consider doing chromatin accessibility and gene expression on the same single cell. Another way is, again, QTL, where we want to understand regulatory relationships. So, enhancer promoter interactions. Obviously, this interaction can be very unique to that individual cell. So it'd be very hard to merge two different information into two different cells into one. So you want this information from the same chromatin. And also promoter DNA information can be critical. And that is because, when you do a bulk DNA methylation, some people say, oh, it's a low methylation. But if you do single cell DNA methylation, you see that there's a high variation in methylation levels. So you have some DNA with high DNA methylation and some cells with low DNA methylation. But in average, you may think it's low, but in fact, there's high variation. So understanding this is critical. Also, expression of protein and RNAs are not always on par. Meaning, although the central dogma is that RNA translates the protein, the expression levels do not always match. So having, again, a protein measurement and RNA measurement on the same single cell will be critical. And this will also be important for patient stratification, because if you can get additional information about the cell, you may be able to understand the patient information better. So overall, multi-omics provides higher accuracy of data and biological interpretation. Yeah. So what are some of the examples of single-cell multimodal technologies? There's a review paper from Fabian Fies's lab, and you can see for genome, there are multiple approaches. So there's even a triple omics where you can profile genome, methylome, and transcriptome all in the same single cell. And there are other variations of this called SCNome, which does the methylation and chromatin architecture, so a taq-seq and RNA at the same single cell. We have other examples here where you have GT-seq, so you have the genome and transcriptome-seq, or MT-seq, methylome and transcriptome. And then you have the RNA proteomics like site-seq, which allows you to measure the protein and the RNA at the same time. And again, there are another great review from Rahul's lab, where they also looked at various methods that are available to do multi-omics approaches. And I'm not from 10x Genomics. I'm not promoting these products at all, but 10x Genomics have introduced various methods over the years where now you can perform, RNA sequencing and a taq-seq in the same single cell. And you can also do immunoprofiling, like a site-seq, where you can do antibodies targeting a cell surface protein. and measure the protein level as well as the gene expression. So here is an example of how chromatin accessibility can be extracted at the same time the RNA, which I'll get into later how this can be achieved. So I have, in the next few slides, I have all these diagrams that shows you how these technologies work. And it'll take a lot of time if I have to explain every single one of them. So I will not go through all of these technologies, but I will direct you to this review paper from Hwang's lab in Korea, where they summarize very nicely all of these methods in detail with these diagrams. And I just want to emphasize that there are really different flavors of multi-omics technologies, and they all have its strengths and weaknesses and their throughput capacity and so on. So I cannot suggest which is better or worse. So what I would highly recommend is to read these and study individual methods and see whether this is something that you'll be interested in exploring. So here is an example of 4 DNA RNA profiling methods from same single cell. Here is another example of DNA methylation with RNA. And again, these are the MT-seqs and then the NMT-seqs. So this is very interesting because you can use methyltransferases to methylate open chromatin regions. And that allows you to identify open chromatin regions and DNA methylated regions. And then you also do RNA sequencing. So again, you get the accessibility, DNA methylation, and transcriptome called NMT-seq. You have a method called SciCAR, and this allows you to profile chromatin accessibility with the transcriptome. And the key feature of SciCAR is this combinatorial indexing strategy. And I will not go into it in detail. However, you are able to barcode cells twice in order to make individual cells a unique feature. And having these barcodes twice, you can identify that's from a single cell. And now you can also profile its chromatin accessibility and RNA sequencing. I would say this approach is probably the most cost-effective way because this does not involve any equipment. You just need a 96 well plate and 96 barcodes and another 96 barcodes. So it's very easy to implement approach, in my opinion. However, it may not necessarily have the highest sensitivity in detecting the gene expression. SNARE-seq is a droplet-based approach, and I believe TEDx Genomics have implemented a similar strategy where you can capture a chromatin nuclear suspension and perform TN5 chromatin accessibility segmentation. And you then conjugate this to a barcode, which is attached to beads. And then you are able to capture the chromatin that has been segmented and capture the transcript that has poly A tail. They can split and sequence both the ATAC-seq and RNA-seq. And again, another multi-modal approach of protein plus RNA. And I think seq is quite commonly adopted, where you have an antibody that targets a specific epitope on the surface of a cell. And this antibody is conjugated to a DNA barcode, which we know, which we can allocate to a specific epitope. And because this barcode can be variable, we can multiplex up to 30 or 40 different antibodies, or even more, I believe, with unique barcodes to perform protein analysis as well as a transcriptome analysis, because this part can be sequenced with your RNA. So I mentioned a lot of the technologies, and again, I highly recommend you to read this review paper. But all of this requires another challenge, faces another challenge, which is how to integrate all of these approaches. The most standard approach would be, I guess, you perform correlations, right? So for example, you correlate the protein expression with the RNA and see whether you can find the pattern, or you correlate the DNA methylation levels to mRNA and see whether there's a negative or positive correlation. Or you can perform a little bit something more sophisticated. And I believe the SURAT3, which I'll get into later, does this very nicely, called anchoring approach. And what they do is they create a reference using the transcriptome data, and then they query using DNA methylation data or ataxic data or other data to compare to its reference and see where they fit in this tSRE plot, in this cluster. So they call this anchoring because they try to match very similar cells from the two data sets, and they anchor the data plots onto the reference. Another approach is, I won't get into much detail, is called non-negative factorization, which is a way to reduce your data and find a feature that is common between the two, and that you use those features to integrate the two data set. And there are, Liger and MOFA are just examples of platform analytical tools to integrate the two multi-omics data set into one. But I don't want to overcomplicate things. So what I think you may do this in your tutorial, but if you have the time, of course, I would highly recommend you to visit this website from Rahul's Satija's lab, where they have a very nice tutorial on how to perform this type of anchoring data integration, not only across different modalities, but even across different samples to minimize batch effects. So again, they do a very good job of explaining things and step-by-steps of how to run them in R. So in the remaining time, I just want to, I don't know how much time I have now. Patricia, do you know how much time I have left?
Yes, you still have some 10 minutes, Jay.
10 minutes, all right. So in the remaining time, I would like to give you some examples of how we're doing multimodal approaches in my lab. And this is a little different from all the examples I've highlighted to you thus far. And the reasons will come very clear very soon. But our question that we are really exploring is how genetic variations contribute to disease, drug response, or even developmental processes. And we do this by studying the non-coding regulatory regions of the human body, or human genome, right? Because the non-coding regulatory regions have the highest number of genetic variations that are associated to GWAS SNPs. So although the protein coding genes are important, these upstream regulatory regions may have a critical role in how gene expression is made, and therefore profiling these non-coding regulatory regions are critical. And we can do this either by ataxic, again, chromatin accessibility, or I'm going to introduce to you by detecting these enhancer RNAs. And because enhancers that are away from the promoters also have a polymerase II activity, and these enhancer regions can transcribe an RNA, and we call that an enhancer RNA. And we can use that to detect accessible regions upstream of the promoter and be able to associate these together is one of our goals. So, so again, the human cell atlas, we're building a periodic table of different cell types. But how are these cell types regulating one another? Again, going back to my very first title page of how the traffic controls and traffic lights can regulate a gene, regulate your driving. So here, how can these non-coding regulatory regions regulate your gene expression? And again, there are very complex activities that are happening just outside of the protein coding genes. And we use what's called 5' end strategy to capture the transcriptional start sites of these promoters, as well as these non-coding regulatory elements such as enhancer RNAs. So our goal is to unveil these Cis-Regulatory elements, we call them CREs, where you have the open regions, and then you have the closed regions, and then you have open regions. And we want to identify these enhancer regions, how they are regulating these promoters in cis, thus the Cis-Regulatory elements. Once we identify where they are, we can then do association studies to correlate how the GWAS SNPs can influence the gene expression, or we can perform EQTL analysis. As I mentioned again, these genotypes can be also present in enhancer regions. So how can the genotypes in enhancers influence the gene expression? We also want to study how these are interacting with each other, physically interacting with each other through Hi-C experiments. And then how we can study these interactions by functional validations using CRISPRs. We can block these enhancer regions and observe how the promoter activity is dysregulated. And we can study all of this. And this is kind of our goal, to do this all at the single cell resolution and be able to build a regulatory map of every single cell type. But today I will mainly talk about the first two examples of how we are identifying them and how we are associating them. So the story goes really way back in 2014 or even longer, where FANTOM is an international consortium where we profiled many different cell types in bulk at that time. And we used this technology called CAGE, or CAP analysis of gene expression, which it's like an RNA sequencing, but it can profile only the 5' transcriptional start sites. And by defining where the transcriptional start site of the gene, we can figure out where their promoters are, where the transcription factor binds, and then it activates the gene. And we can really accurately infer that. But by accident, we also realized that here downstream of this EGR1 promoter, we saw these bidirectional patterns by CAGE technology. And this bidirectional RNA transcription signify that they were actually at the enhancer regions of the DNA. So we are actually using this 5' CAGE technology, and by identifying these bidirectional patterns, we are calling them, that is an enhancer. And then if they have a very strong unique peak here, that is a promoter. So even using the same technology, we are able to identify the promoter and the enhancer. And we achieved this at the single cell resolution a couple of years ago, where we are able to profile these enhancer RNAs in A549 lung carcinoma cells, and that we are able to identify that even at single cell resolution, there's this bidirectional patterns, and we're able to detect this very accurately. So we wanted to also do this in using 10x Genomics platform. And 10x Genomics provides the 5' transcriptome kit as well. So when we do this, when we run the 5' technology, we can get a very nice transcriptional start sites of a MYC loci at the 5' end. But of course, if you use the 3' kit, you get a 3' gene expression pattern here. But we knew, okay, this is nice that we can get promoter expression. And when we compare with the 3' and the 5' gene expression analysis, the correlation is very high. But the question is, can we identify this enhancer regulatory regions. So here's another example of NR4A2 transcription factor. So this is a transcription going right to the left. And at this 5' end, you have this strong transcriptional start site, which aligns very nicely to chromatin accessible regions defined by this red color here. And this is a chromatin accessible region defined by the Epigenome Roadmap Consortium. So we know that this is a proper TSS. But then what about these red regions that are upstream of this NR4A2? Are they regulatory regions? And in fact, when we zoom in to this particular region and scale down, we are able to find this bidirectional transcripts. So now we are able to use 5' single-cell RNA sequencing from 10x genomics to also define enhancer RNAs that potentially be regulating this NR4A2. But interestingly, not all open chromatin regions have enhancer RNA activity. And this is something very important that I'll get into later, that not all open chromatin regions are enhancer active. So we kind of define these CREs, we call them, the Cis-Regulatory regions, defined by T, so T transcribed CREs. And then proximal are promoters, and distal are mainly enhancer-like. And even with 10x genomics, we can observe this bidirectionality at the single-cell resolution. And then we want to understand how they compare to accessible CREs. So accessible CREs are defined by a taq-seq, and then transcribed CREs are defined by 5' RNA sequencing. And obviously the taq-seq is the ground truth because all open chromatin regions should have RNA. And we can see that accuracy is quite good, although it's not as good as the accessible regions. However, if you combine which of the enhancer RNA are interacting with a promoter RNA, in other words, which CREs that are transcribed have a physical high C contact, tSRE has a higher accuracy than accessible CREs, meaning if you use the tSRE as a proxy for interacting promoter and enhancer, you're more likely to find that to be the case if you perform high C experiment, and less likely if you do a taq-seq. So here is a example of how we can study this co-activity pattern. So here's a protein coding gene called PTGER4, and this is a promoter. And then upstream of this gene, you have many of these tSREs, and we can use coactivity analysis to associate these two as an interaction. As what I meant earlier is that these interactions are more likely to be observed using high C than the taq-seq based interactions. And again, if you zoom in closely here, you can see very much 5' RNA-seq expression activities, which are defined as tSREs. So this is going to be a very busy slide, but what I want to highlight here is that these transcribed tSREs, let me give you an example here. These tSREs, these regions in your genome, these are non-coding gene part of your genome, are usually full of SNPs that are associated to a disease. So in fact, Crohn's disease, SNPs are highly enriched in these enhancer regions, but not in this promoter region, right? So suggesting that this SNPs regulates the enhancer activities of PTGR4, and therefore we should be studying and understanding this regulatory region rather than trying to understand this protein coding gene. So this type of association of identifying disease traits to an enhancer is what I summarize in the next slide here. And again, the long story short is that the tSREs can accurately identify disease traits that are associated to immune cells. So here, this is a PBMCs, so we can identify many immune disorder disease highly enriched in this tSREs. And if these accessible regions have high transcription activity, they tend to be much more accurate in predicting the traits. In other words, they can be open chromatin, but if there's no RNA activity, they're not very accurate. But if it's open chromatin and there's a lot of RNA activity, thus enhancer RNA, those type of enhancers tend to predict these disease traits much more accurately. So again, another approach is EQTL. This is very much ongoing, so I will kind of skip over that. So I know what I talked was quite busy, but I just want to highlight that you can actually go to this website and you can explore everything that I talked about. You can actually explore these interactions between the tSREs. You can understand to interpret these variants. You can also look at many other TSS activities in PBMCs if you go to this website. And there's a very handy way to explore single-cell RNA-seq data. These are some of the examples. So in conclusion, I think, I hope that this 5' single-cell approach can reveal functional cis-regulatory elements along with gene expression. So again, there's a very different way of looking at multi-modality here. Although we are only measuring the RNA, we can measure both gene expression and cis-regulatory regions. And because we're using, kind of 10x, which is highly standardized platform, and we are using across multiple laboratories, we can interoperate and relatively cost effective because we can do this in large scale and large numbers. And also we're now multiplexing multiple individuals. So it's going to be reducing the cost. And many of the analytical tools are readily available. So hopefully everyone can be able to do this quite easily. So with that, I just want to close and thank Chung Chao Hon and Jonathan Moody and Kono Tsukasa, who drove a lot of the 5' story. And I'd like to thank everyone in my lab. Just want to highlight, I have a couple postdocs from Latin America, from Peru and Colombia. So if anyone is also interested in coming to Yokohama or Japan, please feel free to contact me and be happy to explore how we can work together. So with that, thank you for your attention.
Thank you, Jay, for this amazing talk. Thank you so much for being here with us today. For those who are not aware, Jay's talking from Japan, so it is a privilege.
It's midnight here.
To have him there. Yes, yes, yes. So thank you so much. We have quite a few questions from our participants, so we're gonna go with those, okay? First from Luis Felipe Antunes Branco. So great presentation. He's asking if it's possible to use PBMC samples frozen for a long time, or frozen in general. I'd say that this could be the first question.
Yes, that's a really good question. And the answer is yes. And especially for our study, when we are multiplexing samples from 16 different individuals, we have to freeze the samples because it'd be very hard to coordinate sample preparation from 16 donors if they're fresh. So we've done some systematic comparison of fresh and frozen PBMCs, and we do find very little difference. And I think it does not, the difference wasn't so big that it would impact the underlying conclusion. I think there are some risks that you may lose some cells, neutrophils or some granulocytes that are very vulnerable to stress. So some of the subsets of the PBMCs can be lost through freezing, but we find that to be very little impact. I think the more critical, is which protocol you use to isolate PBMCs. And if you look in literature, there are, various ways and different coagulants you use, heparin or citrate. And I think those have greater influence in your cell quality and gene expression and cell population. Some isolation can enrich monocytes more than T cells, and some isolations does the opposite. So again, I would highly standardize this if you're planning on collaborating with multiple labs. So they all use the same protocol. So we have done this, and if you want to use the same protocol that we're using, we'll be happy to share exactly what we're doing.
There's another question here also talking about data collection and how important it is. So you made some great points on data collection and the importance of that, I assume. Is data in a Humans Atlas also separated according to age and sex as references for different definitions of healthy?
I would say we're not there yet. I think eventually, yes, because in the end, the metadata regarding age and sex, I don't think every data will have that, but vast majority will. So in the end, once we have all this metadata, we can actually start to ask these kind of questions once they're integrated. So is there a difference between male and female? Is there a difference between adolescents to adults to aging? And I'm sure we will find very interesting data. So yeah, let's see. I think definitely that's in the mind of everyone involved in this project, yes.
Great. Another question from Tiago Lubiana Alves. It is great to see the progress towards standardization. In that direction, to which extent ontologies like Uberon and cell ontology are used for the metadata recordings?
Yes. So Human Cell Atlas has also a huge efforts to annotate the cells. Peter Konchenko is one of the scientists leading this effort. And the goal there is to, of course, generate a tool that can best annotate the cells that is agreed upon many people as possible. So the problem with cell ontology is that some people have just different opinions of what cell is. Some have their favorite markers, and if they express that, that's cell type A. If they don't express that, it's not the cell type. So if you are trying to build a Human Cell Atlas and you have different opinions, it's very difficult to come up with a consensus. So Peter Konchenko is actually trying to build a tool that would allow experts to come together and come up with a definition and building that ontology as a community. So I think it's still in the process of development, but hopefully that kind of approach will provide everyone some satisfaction of coming up with accurate cell ontology.
So within the
Yeah,
Tiago is happy here with the answer. So there is an effort within the Human Cell Atlas to create a consensus, at least for people who are following or are part of the consortium.
Yes, and I think the first step is to create a tool to allow to do that. And I'm probably not the best person to talk to, but yes, there will be a way, a tool for people to provide their inputs, and kind of like a voting or ranking system that will allow cells with the highest accurate annotation or favorable annotations will be kind of ranked higher, and they'll be annotated as that cell type, for example. So I think the best way is through the power of the average, right? You collect information from many people and see who come up with the most accurate annotation.
At least for some time, right? Until something better comes up.
Yeah. Well, I mean, there are many annotation tools right now. There are a handful of excellent sound annotation tools. So, are they accurate or not? It depends on who you ask. So, yeah.
Most definitely, yeah. There is a question here about funding. So thank you for this talk. Could you say something about funding for working with healthy tissues?
Oh, yeah. It's very much a challenge here in Japan as well. A lot of the shifts are going to disease. So, and a lot of our collaborators are clinicians, and they also tell me not just funding, but getting healthy tissues are challenging. So how we go around that is, when we write grants, we do write a very disease-focused grant. It's not big, but very specific disease grants. And in the application, we definitely ask for healthy control samples to be profiled as a reference. And that's how we've been doing it. And because our lab is like a genomics lab and we collaborate with many different clinicians, when we gather disease samples and their control samples from multiple tissues, we can actually correlate the healthy samples and build a small reference. You know what I mean? So it's a little,
I guess that's the solution. Yeah.
Yeah, it's a little going around a bit, but it's a way to collect healthy tissues. As long as we are standardizing the approach and telling we're going to use this technology and this platform, it allows us to not only profile the disease and healthy, but also collect healthy samples in the process. So hopefully there'll be some way someone who can coordinate a lot of these efforts can help you to build that.
Yeah, we here have a lot of difficulties for financing basic research, like pure basic research. So I think we all go around the same way that you're saying. Yeah, that's what we do. Yeah.
Well, so there's CZI, of course. So I would encourage you guys to apply CZI.
Yeah, but everybody, like once they cannot help everybody, I guess. Yeah, of course. Let's hope for more CZIs, right?
Yes.
Yeah, that's what we need. Yeah. So another question here from Juan Sebastian Arcila Barrera. Great talk, Dr. J, about the multi-omics approaches. Is there, I think he means different sensibilities considered methods. Considering the methods, he means like maybe one individual method would be more significant than another one to detect a specific analyte. If you could say something about that.
I would say absolutely yes. Every method has its own strengths and weaknesses and different levels of accuracy and different levels of sensitivity. And accuracy, what I mean by it is, are they reproducible and do they correlate very well across different samples if you do this multiple times? Are they sensitive, meaning can they detect all the genes or detect all the open chromatin regions? And they all have limitations, especially when it comes to single cells, right? When you do genomic, you only have 2 copies of DNA. So trying to sequence 2 copies from single cell, you will suffer from sensitivity from the get-go. So that's very much expected. Which one is better, worse? I'm not here to judge, but I would encourage you to review individual methods and see first, is it feasible in your lab, and second, do you think it's something that makes sense? Yeah.
Okay, regarding that, there is a person here talking about, well, you did show a lot of approaches, right? And how much are them being used?
Okay, well, that question very much correlates to the technology accessibility. If you were to ask me which single-cell RNA sequencing are used the most, just unimodal RNA sequencing, I would say 10x genomics. And that's just because it is highly accessible and it is, the platform is standardized because it's manufactured. So they have high quality standards. So therefore it's expensive, but it's widely used. Now, if I've shown many examples of multimodal, and one of them actually is single cell RNA sequencing and ATAC-seq, and 10x genomics happen to provide that reagents and kits to do that. I don't know for sure exactly if that is the most used multimodal platform to this date, but I can easily envisage that in the next few months or a year, that such platform that is widely accessible and commercially accessible will be most widely used.
We have time. We don't really have time, but we're going to have just one more question here for you before we move on, because Helder is already here and we are right on time. So how do you manage to identify what fraction of the expected immune repertoire in Asian populations was captured by the IDA project? What kind of experimental design leads to best results for this? So I think we're very much interested in this project because we have a very similar structure for Latin America. So if you want to engage in this, what can you say about it?
So is the question like how comprehensively can we profile the population, in other words? Yeah.
Yes, considering this diversity that you have.
Absolutely. There. Yeah. We are doing our best, and it's limited to funding, and again, technology accessibility, in fact. So we want to reach out to every corners of Asia, but first we need the funding to do that, and second, they have to have the capacity to perform single-cell RNA sequencing in their own country. That is one of the aims of IDA, is to ensure that we're not just taking their samples and sequencing it in Japan or Singapore. We want the people in Thailand to collect their own samples and analyze their own samples. So we want to ensure equity is reached. So to do that, we need to raise awareness and raise funding so that we can be as comprehensive as possible in profiling the genetic diversity of Asia. So,
all right, I think that's very much
doing our best.
Yeah, I think that's your experience in Asia is a model for us, because we have the same issues in Latin America, and one of the main purposes of this workshop was that, right, to be able to include different countries and train people locally, to be able to perform part of what we can do, right? Yes. Jay, was really, really great. We're very thankful to have you here. We're not going to hold you anymore because we know it's time to go, right? Yes, time to go. Thank you so much. We're looking forward to talking to you soon again. Thank you.
Thank you,
Jay.
Patricia. And thank you, everyone. Have a good day.
So we're gonna move on to the next speaker, Dr. Helder Nakaya. So Helder has a PhD in molecular biology with extensive training in bioinformatics. Together with Dr. Bali Pulandran, they pioneered the field of systems vaccinology, an interdisciplinary field that combines systems-wide measurements, networks, and predictive modeling in the context of vaccines and infectious diseases. Since then, he has developed systems biology approaches to understand and predict the mechanisms of a broad range of human diseases. And his lab is also focused on applying machine learning methods and single-cell analysis to investigating the basis of infectious and inflammatory diseases. Dr. Nakaya, thank you so much for being here.
Thank you so much.
You will have our full attention now.
Thank you, Patricia. Hey, everyone. It's such an honor to speak here because I saw the level of the talks and what you guys been watching, and that's very, very impressive. So I hope I can talk a little bit more about the tSRE and UMAP plots. I will share my screen so we can better see what I'm planning to. 1 second. Okay, guys, so I hope you can see my screen well. The title that I gave for the talk today is Extracting Knowledge and Insights from Public Available Single Sample RNA-seq Data. And what I mean by knowledge is the pure definition of knowledge, which is the information about a subject that you get by experience or study. So basically, guys, so you all know that in order for us to get knowledge, we need data, right? So we need to do analysis with this data to learn about what we did. And to collect the data, we need to do the experiments. At least the scientific knowledge is acquired this way. But the thing is that it looked like that modern biology fits what Asimov said once, which is the saddest aspect of life right now is that science gathers knowledge faster than society gathers wisdom. Of course, he was not mentioning single-cell transcriptomics, but what he's saying is that we are really good in producing a lot of knowledge, but knowledge by itself means nothing if it cannot be translated into understanding and wisdom. So what I want to focus now is not only on the data analysis, the normalization, but how can we do this conversion of knowledge into understanding and wisdom? And what do I mean by that? So for example, we can run an experiment, which is like get eggs and put it into a pan with boiling water. And we can repeat this experiment 300 times, so we are gonna have a lot of data. And we all know what's gonna happen. We all know that the eggs are gonna be boiled, but very few of us will understand precisely what's going on at molecular level in these boiled eggs. So that's the deeper understanding of what's going on here. Same thing with single cell data and single cell analysis. For example, we can take subjects that have COVID-19 or healthy subjects or people that die for another cause, and people can do like an amazing work, which is profiling at single cell level several different organs of individuals that die with COVID. So there is one paper that came out yesterday that shows some of this profiling from the organs of autopsy organs. So once they do this huge data analysis, which you all experience in this workshop, and then you're gonna do the hands-on later, is that, for example, one of the findings was that CTHRC1 positive pathological fibroblasts may be contributing to the rapidly ensuing pulmonary fibrosis in COVID-19. Okay, but of course, in one paper, even if it's a Cell paper with 8 figures and 10 panels each figures, you cannot cover everything. You cannot understand the whole thing. You can't simply identify all the pathways and genes, transcription factors that are involved in COVID-19 in different organs. So even in this paper, they finished by saying that this atlas established an important resource to investigate host response to SARS-CoV-2. So this provides a basis for therapeutic development for severe disease. So that means that we are acquiring more knowledge, and this knowledge becomes a resource for understanding and wisdom. So as I said, what you guys already had a lot, and in a top level, is a lot of talks that talk about this, that shows all the technology, all the wonderful things that you can do. And I confess that of course I get jealous because in Brazil we start generating data, single cell data, like very recently, like few years ago, like one. So when we are now generating single cell data, people are already doing single cell of the whole mouse, you know. So it's but the point is not to talk about these things, but it's to talk about these parts here. So how can we use actually our scientific brains to convert data into knowledge and then understanding? So I think that this could be a useful way for Latin American researchers to start already learning to extract knowledge from the existing single cell data. So because people cannot simply analyze or extract everything that it can from one single experiment. So in the past few years, we have been applying systems biology and network medicine on several different diseases, especially infectious diseases. So malaria, influenza, yellow fever, Ebola, Zika, dengue, Chagas. And also we are now looking more into neurological disorders and inflammatory disease. So, but most of these papers here were done with bulk RNA-seq, but these are the examples that I can tell you we can apply also for single cell. I'm going to show several examples of how we could do this for single cell data sets, but the way we learn in the lab of working, which is the same we are applying now for single cell, was on this type of disease. And of course, when the COVID-19 pandemic hits us, we focus our efforts. All the lab was doing different projects on Ebola, on COVID-19. So from app to diagnose to epidemiology to computational tools to systems biology. So of course, I'm not going to talk about all of them, but I'm going to talk about the ones that use single cell data. So we learned about how to extract knowledge from when I was in Bali Pulender's lab at Emory University as a postdoc when I started that. So what we were investigating that time was why the same vaccines can trigger very different response to people. So why some people have adverse events to the vaccines, why some people do not respond at all to the vaccine, and some will have a very high antibody response or CD8 T-cell response. And the tool at the time was extracting the blood from these people before and after vaccination, and then measuring the bulk transcriptomics. So we have the mix of all cells and the transcriptome from this. So the question that always appeared since 10 years ago was, how can you extract knowledge from this huge amount of data that come out from this kind of experiments, right? So which cell, which pathway, which gene I should choose to go deep and understand more about the mechanism of vaccination or the mechanism of infectious disease. So when you do like a bulk analysis, you find tens of thousands of genes and hundreds of pathways that you could look into. So it's simply impossible if you randomly select one of these genes or the pathways to perform more experiments, to learn more about the disease. So what bioinformatics and computational biology can do for you is not to provide the single gene or the single pathway that you should pursue and put in the title of the paper and do a lot of experiments, knock out mice. It cannot give you this. So the only thing that it does is like a funnel where you take a lot of results and then you use these tools, these pipelines, these scripts to reduce the amount of things that you as a scientist have to investigate. So at the end, you still have to use your expertise, your knowledge, previous knowledge about immunology, about the disease, about the vaccines, and also, of course, even your intuition that what could be cool to look at. But the computational tools, the pipelines that you all see during this workshop is just a way of reducing this amount of information. And then after you do this exercise, you can ask yourself like, okay, so which gene or pathway I should focus on? Should I focus on the hub gene, which is actually not the one which is the most differentially expressed, or should I try to look at the differentially expressed genes? Or I can use the module of the genes to look at as collectively what's going on with my perturbation, for example, right? So in this case, when you perform this analysis, you still end up with dozens of genes and dozens of pathways. Okay, guys, so keep in mind that workshops like this are amazing and necessary, but at the end, what you are gonna get is a smaller set of biological entities to look at. In this part, there is no program, there is no button, no pipeline that you press a button and you come up with the understanding or the experiments that you have to do to validate that. So what example I started from this paper here, where we vaccinate people with yellow fever vaccine and then measure the immune responses, antibody and CD8 T cell response. And the signatures, and one of the signatures that could predict the CD8 T cell response was this one. So we found dozens of genes, I'm just showing some of them, that were that we could use the expression levels to predict who's going to have a high CD8 T cell response or a low CD8 T cell response induced by vaccination. And then the question, guys, is which one should we choose to perform knockouts experiments, to do like validation, the biological validation necessary for really understand the phenomenon? So we could choose, for example, this gene here, we could choose these ones, or we could choose these ones. Which one should we choose? So we chose to look at GCN2. Actually, I'm telling this not the whole story, because the whole story, I looked every single gene and I went to PubMed, read a lot of papers, see if this makes sense. If it makes too much sense, this is bad, because that means that this is nothing new. But if it's something that it's something, a good insight that this could provide a new mechanism, that could be nice. So GCN2 was a gene that is is related to, is a kinase that is related to amino acid sense deficiency. So this gene is part of the integrated metabolic stress response of the cell. It's a very ancient gene. So it's a very cool gene to study, and it detects stress of the cells. And also there were at the time some evidence that GCN2 was involved with 1 or 2 viral infections. So maybe this GCN2 could be somehow related to the CD8 T cell response. And that became the postdoc project of Rajesh and Noor, that showed that the yellow fever virus could trigger starvation of the amino acids, that trigger several genes in dendritic cells. So these genes involved the transcription of several genes related to innate immunity, and that affects autophagy, the inhibition of translation, which is very common to several virus infection. And the yellow fever vaccine is actually a live attenuated virus vaccine. So there is a replication there. So what they show using knockout mice experiments in vitro was that GCN2 was important to trigger the autophagy in dendritic cells, and that was related to the antigen presentation to T cells. And the T cells explains probably why it was important for mounting a good cytotoxic CD8 T cell response. Of course, they did a lot of experiments to show that there was an unappreciated link between virus-inducing integrated stress response in dendritic cells and the adaptive immune response. So that paper appears 5 years after the Cis vaccinology publication. So that means that it takes a lot of time to learn and go deep and understand the phenomenon. But we could only choose GCN2 because of this initial paper. So Rajesh was using the GCN2 knockout mice, and he noticed that when you you make acute colitis models inducing with this DSS, the knockout mice had a more inflammation. It has a change in the guts. So he was wondering that what is the implication of this to humans? I was already in Brazil with my lab, so he asked us to look at public available data to see if GCN2 was changing in ulcerative colitis patients. So I asked my students to go into GEO to find studies of guts, bulk, transcriptomics, and then he found that actually GCN2 was highly expressed in duodenus, in cecum, and that there was an increase of GCN2 in patients with ulcerative colitis and Crohn's disease compared to health controls in 2 studies. So you see, like we do not need to recruit patients, go to ethics committee, do the experiments, and then get this data here. We could only go directly, if we have the right question, we can go directly to the database and try to answer these questions. So that became part of this Nature paper. So Leonardo helped a lot here, but what they show is that GCN2 controls gut inflammation by inhibiting inflammasome activation. So since I joined Bali's lab back then, we focused on understanding why the same flu vaccine acts differently in children, young adults, or elderly. So what I want to show is extracting knowledge from systems biology data sets. So in this paper here, we identify signatures that could predict who's going to develop a high or a low antibody response. So there were dozens of genes here. So I look at every single gene, go to PubMed, go to Google, try to read thousands of papers, and then I come up with some of the genes that I thought could be nice to go deeper into validation. So one of the genes that I selected was CMK4. CMK4 is an enzyme that is also a kinase that is implicated in lymphocytes, neurons, and male germ cells. So there was already a hint that CMK4 could be involved with antibody response because someone showed that CMK4 was more related to T cells at the time, but nobody knew what is the role of CMK4 in the B cell response, the antibody response. What we could see is that when you look at the antibody response and the expression of CMK4 after vaccination, there was a negative correlation here. So people that have higher expression of CMK4 were the ones with the lower antibody response. The people with lower expression of CMK4 were the ones with higher antibody response. So the experiment was, let's get now a knockout mice for CMK4. So the mouse do not have CMK4, and let's measure the antibody response. So this is the antibody response of a wild type mice, you see? Because CMK4 is negatively correlated, we expect that a knockout mice that do not have CMK4 would have a higher antibody response. So when we immunize the mice, the knockout mice, we do see that CMK4 knockout mice had more antibody response. So what about the lymph nodes? What about T cells, B cells? So we could, at that time, look at the lymph nodes, for example, of unvaccinated mice. You have to look at the green dots, which are the cells that produce the antibodies. And then this is the lymph node of a mouse, a wild type mouse at day 4. You do see some green dots here. But when you do the same thing with the CMK4 knockout mice, the lymph node is smaller, but you see that it's full of B cells here. So that was part of this paper, but we also found more stuff that could be later extracted more deep into understanding. And one of the figures of this paper here was this one here. Remember that I said you have fewer genes to choose, but still you have to look at every single gene or cell and decide if that it is important for follow-up experiments. So it's different than you just describe as an atlas what are all the pathways, all the genes that are changing in all the cell types. That's this is a description of what's going on. Here we are trying to select what we are going to do for moving forward with the understanding. So one of the genes that we thought was interesting was toll-like receptor 5, TLR5, because TLR5 was positively correlated to antibody response. So that means that if you have more expression of TLR5 after vaccination, you have more antibody response to the influenza vaccine. And that is interesting because toll-like receptors are important innate immune receptors. So somehow this could be a nice insight about the vaccine. But then Rajesh quickly told me that was bullshit. That is probably a noise or an artifact, because TLR5 should not be there as important to a vaccine which is composed of an inactivated virus. And why? Because toll-like receptor 5 binds to a bacterial component, flagellin. So flagellin has nothing to do with inactivated virus vaccines. But then again, like I read several papers that shows that somehow there were, for example, examples of groups that use flagellin as an adjuvant for flu vaccine and that worked. So the TLR5 was related to other stuff. So let's do an experiment. So Jason took the lead, and the first experiment that he did was to get a TLR5 knockout mice and immunize with the flu vaccine. As you can see here, guys, the antibody response in the knockout mice is lower than the wild type mice, meaning that, I don't know why, TLR5 was important for antibody response. So the next question is, okay, so what is binding to TLR5? Because that is definitely not a contaminant in the bacterial contaminants in the flu vaccine. So where the ligand was coming from. So that, guys, was 2011, 2010. Nobody talks too much about microbiome. So the hypothesis at that time is that maybe there is, it's coming from the commensals in the guts. So that's what motivates me to do science. These kind of like quests of trying to understand why this is happening. So the idea is that maybe it's coming from the bacteria in the gut. So how do you test if the bacteria in the gut is important for antibody response? Well, if this is true, if you can treat mice with antibiotics, and then you're going to see that the bacteria in the feces, in the stool, decreases 26 times if you treat mice with antibiotics. Or you can even buy germ-free mice, and you're going to see that there is an even greater reduction of bacteria. And then you can immunize them and see if the antibody response changed. You see that if you treat normal mice with antibiotics, or if you buy germ-free mice, the antibody response is diminished, the same level as the TLR5 knockout mice. So that shows that the flu vaccine needs the help of gut microbiota to mount a good antibody response. And that only appears because vaccinology here gave us some of the ideas of testing this later in mouse. And in Bali, I was already gone from Bali's lab. He published in 2019 a paper where he went back to humans, treat humans with antibiotics, and then vaccinate them to show that even us humans, our gut bacteria can affect the immune response. So this is important because maybe if we do single cell RNA-seq in people with different diets, you're going to see a completely different perturbation. This is by itself a very important perturbation. Okay, so what about public available single cell RNA-seq data? So now let's give some examples of how can we do the same approach for single cell RNA-seq data. So this is the paper that was published by Cesar, which is one of the panelists of this workshop. I think he's going to talk next week. And it's about juvenile idiopathic arthritis. So when he was doing a PhD in my lab, he did a massive survey of all the studies related to juvenile idiopathic arthritis. So this is a chronic inflammatory conditions that affects children. But the thing is, guys, that the disease does not affect male and female children the same way. Not even the treatment looks the same if you treat male and females with juvenile idiopathic arthritis. So when he did this survey with hundreds of studies, he came out with some of the studies that he thought could be nice to compare between male and female. So we did a meta-analysis. This is a tool that he developed as well called MetaVolcano. I'm asking him to finish this paper every single week, but anyway, so with this tool, we identify genes that were upregulated or downregulated in children with arthritis, okay? So what he did then was to compare the expression of these differentially expressed genes between male and female children. So these are the genes that are upregulated in female, and these are the genes that are upregulated in boys. So when you do the enrichment here, you see that the genes here are enriched for neutrophils, and then that's fine. Neutrophil seems a very related cell type for the arthritis. This is not new. But then what he did was was taken single cell RNA-seq data from this paper here, where they had over 45,000 cells. And the nice thing is that they screen here progenitor cells, so cells that are not necessarily mature cells that you commonly find in PMBC. So when we reanalyze this data, he identified, for example, 33 cell clusters. And then he interrogates about the genes that were upregulated in females compared to males in this cohort here. And then what he noticed is that some of the genes were enriched in a specific cell clusters of a specific type of neutrophils. So what we learn is that most people talk about arthritis and the mature neutrophils that you commonly find in the blood, but there are like different types of neutrophils that are differentiating to the mature one. The immature forms that are commonly found in the bone marrow, some of them goes to the peripheral blood, and some of them that goes there is these band neutrophils or low density neutrophils. So to learn more about if this is true that low density neutrophils are important for juvenile idiopathic arthritis, he went into this other paper that have signatures of different types of neutrophils. And then he looked at, in these different types of neutrophils, if the genes that were related to the sex differences, increasing in female, in girls, for example, were the ones related to band neutrophils, the low density neutrophils here. You see? So that means that probably what we are seeing as a signal are coming not from the mature neutrophils, but are coming from the band neutrophil ones. So, and then he did some trajectory inference analysis where he took only the neutrophils here, and then he put them in order of pseudotime here. You can see the order of different neutrophils, showing that these guys here are less differentiated in this pseudotime here, and these ones are more. You see, and then he can look at the expression patterns of important genes that explains the sexual dimorphism of the disease in these different stages of neutrophil differentiation. So what interests me at the time was to understand why the treatment of boys and girls are different if you use the same drug. For example, one of the drug is anakinra, which is an inhibitor of IL-1 beta. So if you use the blood expression as a proxy of to measure the activity of those band neutrophils, you find these results here. You see that it looked like that females had a little bit more activity of the genes related to the band neutrophils compared to males with no treatment. Of course, if you treat them with placebo for 1 month, nothing changed. But if you treat boys with anakinra for 1 month, you see that the activity decrease. And if you treat for 6 months, the activity decrease even more. But look at now, guys. If you treat girls with the anakinra for 1 month, this activity does not seem to change. So meaning that probably the band neutrophils could be important to explain these differences in treatment, for example. But we want to know why. So why there is these differences between boys and girls if several of these patients are actually even before puberty. So does the sexual hormones play a role here? So what he did was to take thousands of transcriptomics, blood transcriptomics of males and females over different ages groups. And then he saw that if you use the mean expression of the genes of these band neutrophils here, you see that they are sensitive, they change over age. You see that females and males, they change. So that means that could be related to hormones. And then I asked him like, so how can we measure the influence of hormone without having to do experiments, without having to pay for recruiting patients and treating with them with hormones and everything? So we had an idea of getting public available RNA-seq or microarray data of pregnant women. So because we know that during pregnancy, the hormones change a lot. And in fact, if you look at here, non-pregnant women, the activity is here, and you see that over the gestation periods, the activity increase together with the female hormones. And then it goes back to normal after birth. So that means that probably this has to do with the sexual hormones. So this was published, I think, last year. So you could all see, and I hope that Cesar is gonna at least mention this in his talk. So we also apply single cell transcriptomic analysis for COVID-19 and also comorbidities. We have like actually 5 or 6 people in the lab working with this, but the result that I'm gonna show you was done by Leandro and Icaro. So in one of the papers, we are trying to understand why people with heart failure, diabetes, chronic obstructive pulmonary disease, obesity have higher chances of dying of COVID-19 compared to those that do not have these comorbidities. So what are the molecular factors that are associated with severe COVID-19? And that is important because these are different diseases. So why different disease, they all increase the chance of dying of COVID-19. So this is just a way of showing why I'm talking about this, because the World Health Organization declared a COVID-19 pandemic on March 11. And we submit our paper, we post this paper in March 27. So that's why now it's not gonna look like a completely novel result, but at the time was nice to do that, because we did all this without spending a single Brazilian real, because that was all with public available data. So we went quickly to the gene expression omnibus and looked for all the comorbidities associated with severe COVID-19, which were also done in lung tissues. So at that time, there was not a single cell RNA-seq of COVID-19, at least not available or that we could find. So, and definitely not from lung. So we wondering, what are the genes in these different comorbidities that were up or down regulated in the lungs of these people? And then we use again the metavolcano to identify thousands of genes that were up or down regulated in COVID-19. So this is the metavolcano web tool. We also have a bioconductor package, so you can use this for even combining data of single cell results. But basically the web version allows you to not even know R. You can just drag the expression file to the tool, and then it's gonna show you like the number of DAGs per data sets. So these are the number of DAGs that are in at least differentially expressed in at least 6 data sets. Oh, it's also important to mention that this is also done with Vinicius Maracajá, which is one of the organizers of this workshop. And then you can see the results. You can play with, interact with the results. You can see how many genes or which genes is the most upregulated or the most downregulated in most of the studies. So that's why what I'm saying. These pipelines, these scripts do not give you like what is the insight that you need to explain something. It's only going to reduce even more what you have to look for. So there are like different ways of analyzing the results. But at the end, so what we did was to take the differentially expressed genes, do a path enrichment analysis. And then we found, for example, genes related to viral life cycle. And then we see that among the genes inside this viral life cycle, there was ACE2. I know that everyone knows why I'm going to talk about this, but ACE2 is the main entry, the main receptor used by the virus to enter the cell. So it was nice to see that ACE2 was upregulated in the lungs of people with different comorbidities. So that could explain maybe that people that with very different comorbidities will have high levels of ACE2. So if they are exposed to the virus would easier find, it would be easier for to the virus to find cells to infect. That could lead to increased SARS-CoV-2 replication and also increase the risk of developing severe COVID-19. And that is already known that if the input amount of virus that you receive is higher, you are more likely to have a more severe disease. So that was our hypothesis. And in fact, when we analyze RNA-seq data, we do see that the ACE2 was increased in people that are frequent smoker, or they have chronic obstructive pulmonary disease, or they have pulmonary arterial hypertension. This could also explain. So the next question is, okay, so why ACE2 is upregulated there? So what is regulating ACE2? Again, we did not perform ATAC-seq, we did not perform DNA methylation studies or nothing like that. So what we could do is to look at the expression of different genes with the expression of ACE2 in these studies. So we can measure, for example, what is the level of transcription factor in ACE2 and see if there is a positive correlation or a negative correlation. So this analysis it's called guilt by association. So if we find genes that were very positively correlated with ACE2, that means that probably they share the same transcription promoted regions, they have the same functions, or one may regulate the other. So when we did this for 7 studies, we found, for example, 544 co-regulated, co-expressed genes, so genes that were correlated with ACE2 expression. And when we do the enrichment analysis with this list here, we see, for example, that there were several targets of specific epigenetic regulations. So for example, this H3K4, ME3, H3K4 methylation 1. So what we did was to go to the Rhodes Epigenomics database, and look for these markers in the lung there already, and then focus on the ACE2 locus. And then we found that indeed, the locus of ACE2 seems to be epigenetically regulated by these marks here of histones. So that could probably explain or may explain why ACE2 was upregulated in people with these different comorbidities. So this paper was already published. And what I want to talk is about another comorbidity that is very frequently associated with COVID-19 death, which is diabetes. So the goal here is to understand why diabetes was important for increasing the chance of dying of COVID-19. So I'm gonna tell a very short story because we only participate in this study. But what we did here was to get the single cell data of the bronchoalveolar lavage of the lungs of COVID-19 patients. So we reanalyzed the single cell data and identify which clusters were CD14 monocytes. And I will tell you why we look for CD14 monocytes. But the fact is that there were a lot of monocytes, human monocytes, going to the lungs of people with COVID-19. And when you look at the interferon alpha beta signaling, you see that people that have mild or severe COVID-19 have increased expression activity of interferon alpha and beta signaling in CD14 monocytes. That gives us an idea that probably monocytes could be infected with the SARS-CoV-2. So we did this analysis because the people that have the question was people from Unicamp. Pedro Vieira's team was actually learning that in vitro, if you cultivate monocytes and infect with SARS-CoV-2, there was a replication. But if we put increased amount of glucose, there was an increased replication of the virus. So that was cool because that shows that glucose somehow favors the replication of the virus. And that could also explain why diabetic patients will have a more severe form of the disease. And what we are using here, the single cell data, was to show that indeed CD14 seems to be infected with SARS-CoV-2. But then when we look at other pathways that were related to CD14 monocytes in mild and severe COVID-19 patients, we do see that glucolysis is one of the pathways that were activated there. So what Pedro's team did was to look at, to block, inhibit parts of the glucolysis. This is the fermentation, and this is the cell cycle and respiratory chain. So I'm not going to show all his results, but if you block, for example, this step here, you see that when you use vehicle, the virus replicates in the monocytes. But if you block using this inhibitor, there is no replication anymore. So that means that the SARS-CoV-2 was utilizing glucolysis. This was necessary for the virus replicates, and that involves HIF1 alpha, which is related to hypoxia and also oxidative stress. So this was published by their group here. We also only helped with the single cell part, but they show that elevated glucose levels favor SARS-CoV-2 infection and monocyte response through HIF1 alpha glycolysis dependent axis. So what about other potential comorbidities associated with severe COVID-19 that is not even here? So can we find new comorbidities that nobody thought in looking at, because that has nothing to do with lungs, has nothing to do with vascular problems? So that appears by randomly, because Leandro in my group was investigating this disease called Barrett's esophagus. So when this is the normal esophagus and this is the esophagus of people with Barrett's esophagus. And this happens when you have a chronic reflux or gastritis that change the morphology of the normal esophagus. So this is a condition that affects 5% of US population and 5% of Brazilian population. So Leandro was performing a meta-analysis of 8 transcriptomic studies of Barrett's esophagus. So the first thing he did was to see which genes were commonly upregulated or downregulated in most of these studies. So he found 300 genes upregulated and 256 downregulated. But then when he started looking at the genes that were there, he saw that AC2 was actually one of the top genes, one of the genes that were more highly expressed than any other gene here. So that gives us an idea that maybe the Barrett's esophagus could be a comorbidity associated with severe COVID-19. When we look at the pathways related to the upregulated genes and downregulated genes, we see genes downregulated related to the change in the morphology of the mucosa, the esophagus. But we also see pathways related, for example, with regulation of intracellular pH. That is kind of cool, because people that have chronic reflux or gastritis, which is the acid pH of the stomach goes into other regions, that could explain why the pH appears here as a pathway, enriched pathways. And then we look at if ACE2 was regulated by the acid pH. So there, amazingly, at that time, there was very little known about the expression of ACE2. Where we knew at the time that HIF1 alpha was involved with the regulation of ACE2, but nobody showed that pH could be involved with the regulation of ACE2. So which experiment could we do to see if the acid pH was important? Well, we could pay reagents, go to in vitro and then stimulate cells with acid pH, or we can look for public available data sets where people treat people, for example, with medicine that controls the acidity of the gastrite, for example, omeprazole here. And you show that, and when Leandro reanalyzed that data set, we saw that ACE2 diminished after you treat with omeprazole. And again, several papers related to pH-related gene sets. And then we went to a single-cell RNA-seq data of Barrett's esophagus paper. So we went there because we want to look at the expression of ACE2 in different types of cells in these data sets here. And then we did see that most of the expression of ACE2 came from the duodenum, and that was already known. So, but we do see that a fraction of cells from the Barrett's esophagus have expression of ACE2, and that's, if you take the genes related in these cells, they are also enriched by cellular pH changes. So the question is, does ACE2 expression increase in SPH? So we could go in public available data sets and see if they study where they treat with acidosis or decrease the pH, what happens with the expression of a single gene. And then we found that, yes, indeed, ACE2 was always higher if you have a more acid condition. And then we asked Pedro and Luis to validate this for us in vitro. So what they did was to take human primary monocytes and then treat with normal pH and also more acid pH. So this is the expression of ACE2 in these monocytes, and this is what happens when you put them into an acid pH. That shows the ACE2 increase. And then the question is, okay, so this increase leads to more virus replication. So when we infect the cells here, we see that there is more virus replication in the cells with more ACE2, which is the cells with less pH, so more acid pH. And all this was done in normoxis. So we control for the HIF1 alpha and only try to look for the role of acid pH. So what about the clinical implication? Can this be translated into humans? So nobody, at least at that time, designed a cohort to investigate people that have Barrett's esophagus, gastritis reflux, and then COVID-19. And then how could we test this hypothesis in vivo, in humans? So what we know is that people that have Barrett's esophagus, gastritis, and reflux, they usually take proton pump inhibitors, such as omeprazole. So we could go to the records, the medical records of COVID-19 patients, and then separate the people that were taking the proton pump inhibitors from those that were not taking the proton pump inhibitors. So this is the group that we assumed that were people that were treating for problems with the acid pH. So these are the people that supposed to have Barrett's esophagus, gastritis, and reflux. So when we do the analysis, we see that people that use these medications, they spend more days in the hospitals, and the risk of ICU admittance increases as well. But look, when you look at the survival curve, guys. So this is the survival curve of someone that is not taking the medication, and this is the survival curve that people that take the medication. You see, this is almost 600 patients. And when you control for comorbidities, when you do the analysis controlling for diabetes, hypertension, old age, you still see a twofold increase in risk of dying of COVID-19 if you take this medication. And then we replicate this into an independent cohort done in another region of Brazil, with more people. So in total, we have over 1,000 patients. And then again, we see that there is an increased risk of dying of COVID-19 if you if you are treating for these drugs. So now I'm going to be very quick here. Patricia, how many minutes I have? How long?
Actually, it would be good if you'd be wrapping up. Otherwise, we're going to go too far
Okay,
in
okay.
our
So this is to show that COVID-19 also changed the amount of lymphocyte that you have, and this leads to cytokine storm and immune system dysfunction. So what if SARS-CoV-2 can bind to CD4 receptors and infect T helper lymphocytes? So this is a paper that we have the honor to participate, where they showed that you can in vitro infect CD4 T cells with COVID-19, but not CD8. And if you take the blood of people with severe COVID-19 and sort for CD4, you do see virus there. So what we did was to use single-cell RNA-seq data to look at CD4 T cells, and then we use the viral tracker to see if we could find cells that were infected in the lungs of CD4 cells of people with SARS-CoV-2. And then we found that there was a small proportion, which is similar to what we expect in vivo data that they were showing. COVID-19 also patients also exhibit neuropsychiatric and neurological symptoms. So this is a paper with many, many authors. Of course, I'm not the corresponding author, but what we did was to look at what they did was to infect astrocytes with COVID-19. So they showed that astrocytes in the brain could be infected with the virus. The problem is that astrocytes do not have AC2. So what is the entry receptor? So we took a single-cell RNA-seq data that was not even published, was in the bioRxiv, and then we reanalyzed them and found some putative receptors that could explain the entry of the virus in the astrocytes in the brain of people. So they later showed that the NRP indeed was expressed there in astrocytes using Western blots. And look at nice. So if you infect cells with SARS-CoV-2, astrocytes increase the replication. But if you block NRP1, there is a decrease in the amount of virus infection. So we also helped with single cell analysis, this paper here, where they look for efferocytosis. We also helped this paper here with single cell data for comparing males and females patients with COVID-19. We also use single cell RNA-seq data to investigate neutrophils in sepsis. So basically here we were looking specifically to one gene expressing neutrophils to try to explain if this gene was related to inflammation. And the other story of sepsis, we're looking at plasmablasts in the sepsis and also in the lung to show that actually the genes that were involved in the plasmablasts of sepsis were actually several genes related to the Krebs cycle, mitochondrial genes for the respiration. And what they did in this amazing paper was to, again, block some of these parts to see if this is important for what we are seeing in vivo. So as a conclusion, guys, is that although what I was showing in the beginning was the bulk RNA-seq approach, but with the single-cell RNA-seq approach is very, very similar. The only things now is that you have more data. And if you have more data, you have a bigger funnel. So you need more pipelines, script normalization process. But at the end, you're still going to have more information. And from here, it's basically there is no other pipeline. That's the human expertise. That's the collaboration that you do, the questions that you ask. We also helped some of these two here. So this is the project from Thiago that developed this FCOX package. So basically it was inspired in this other tool that we published, led by Pedro, which is the semi-tool that do correlations. But the problem is that you cannot do correlations analysis the same way with the single cell data because this is a sparse data. So Thiago showed that using information theory and entropy, and he could find the correlation between genes, and that could give you more insights about what's going on. So again, Does not give you the final answer, but at least show the insights. So I hope I convince you guys that there are already plenty of studies and data sets of single-cell RNA-seq. We do have amazing tools you all see in this workshop and the database which are available, and we can do low cost and high quality science. However, this equation is not necessarily direct. It's not that simple. It's not just getting the tools and the data sets and then you're gonna have low cost science. And the reason for this is this story here that I like to mention, which is from the Hitchhike Guides from the Galaxy. And in that, they tell a story about alien planets where they were talking about what is the meaning of life, the universe, and everything. So they were tired of thinking about that. So what they did was to build a supercomputer, the size of a city, and ask the computer, what is the meaning of life, the universe, and everything. So the computer said, look, I'm gonna have to think. And then they're gonna say, okay, how long? And then the computer said, 7.5 million years. And then say, oh my God, 7.5 million years. Yes, that's what I need. So 7.5 million years later, they all went to see the final answer for this question. And then the computer said, okay, the answer is 42. And then everyone's like, what? How this could be the answer of the meaning of life, the universe, and everything? And then the computer said, like, look, I recalculate several times and I'm sure that the answer is 42, but maybe you know you did not ask the right question because the answer for the right question is 42. And then the alien said, okay, so what is the question, the ultimate question? And then the computer said, oh, I'm gonna have to think some billions of years, and never gave the answer. So the message here is that there's no point in having a supercomputer, have an amazing pipeline, have a huge data set, a huge technology, amazing technology, if we do not know how to ask the right questions. So we should never forget about our scientific training to analyze and interpret the results. So with that, I would like to acknowledge the people from my group. So some of them are actually attending the workshop. Tiago is here. So we have people from different backgrounds. So this is the Computational Systems Biology Laboratory. And this is the people from the lab. Okay, guys, sorry for talking too fast.
Helder, that was great. That was a great ending. The right question. That's what matters. Thanks. And one of the reasons you were invited, you touched that like beautifully, which is in Latin America, we don't really have data to analyze so far. We're all starting, but there is a lot of data available. And this is the first workshop that comes as a consequence of last year, right? You were there with us discussing what we could do during this time, why we don't have data, and it is training and it is understanding what this data means and how we can use it. So you did this brilliantly. You showed how we can actually take advantage of that, right? So thank you so much. Thank you so much for that.
Thank you, because I think that's the whole point of why I would like to give this talk, because I know that you guys already have amazing pipelines showing to Latin American researchers, but maybe we can help
you
with
did
this
it.
part.
You did it. It was great. That's exactly what the message that we wanted people to have. So that was very good. We're gonna ask you like some 2 questions because we are a little bit ahead of time. Yeah. Okay. So first one, great talk. How do you expect single-cell RNA analysis, I'd say, to improve results that you have on response to vaccines?
Yeah, I did not show this because I was not part of the study, but I know that, for example, Bali Pulendran, my former advisor, every study that he does now with vaccine, he do single-cell analysis. So there is a preprint, nice preprint of one of the mRNA COVID-19 vaccines where he used single-cell data, site-seq, to provide a nice overview of all the cell types that are responding to the vaccine. So I'm pretty sure that everything that we used to see with bulk RNA-seq, we are going to see now with single cell. So it's very exciting time to do science.
There is a question here about the MetaVolcano tool. So it's Maria del Rosario Livingho. She's asking if it was designed to be exclusively used for idiopathic arthritis, or if it could be adapted to other disease data.
No, Maria, actually, MetaVolcano is designed to any meta-analysis that you want. Actually, even if you have cytokine data, if you have proteomic data, metabolomic data of different disease, it's basically the input files are the fold change, the p-values, and the interval coefficients, you know, so these are, there are some parameters that any high throughput data set generates that you can use as input for MetaVolcano. So I'm glad that you asked this, because Cesar is here. I'm always pressuring him to finish the publication, but you can find him in Bioconductor.
I don't know if Cesar wants to say anything about that, but possibly not. But anyway, yeah. A final question before we go from Sebastian Urquiza. Very good presentation. I wanted to consult you. Long non-coding RNAs have low expression values, and I imagine that at the cellular level, they should be even lower. Do you think it is possible to detect the effect of long non-coding RNAs in response to different conditions or diseases?
Yes, Sebastian, that's a great question. That is true for most of the long non-codings, but we do see, for example, Cis, Malati ones. So several long non-codes that are actually famous ones are very much expressed, even among the top genes most expressed in infectious inflammatory disease at single cell level. So of course, if you want to look for that long non-codes that is very rare, you may not find, but that's the beauty of single cell, right? Because people know that long non-codes are very tissue specific. So with single cell data, this signal is not going to be diluted anymore in the bulk transcriptomics, but because now you may find in a very small group of cells, the expression of the long non-code that you want very, very high. So I think that the long non-code field is going to explode as well because of these techniques that allows you to look for smaller groups of cells and tissues and conditions.
Helder, it was really, really nice to have you here today. Thank you so much for your participation. Hopefully next year, next time we hold this, we're gonna have many more people from Latin America being able to show data just like you do, from practicing and training during this upcoming year. Thank you so much for your help.
That would be great. Thank you so much, guys.
Bye-bye. Hello, welcome back to the afternoon section of our workshop, in which we will have our last two talks for the people participating of the first week of the event. Our first speaker is the Professor Dr. Musa Mlanga. I hope I pronounced well correctly your name. He did his undergraduate studies in France and the US and obtained his PhD in New York at the Rockefeller University and at the New York University School of Medicine. There, he worked on the development of molecular beacons for in vitro diagnostics and for imaging of RNA in living cells with Fred Kramer and Sanjay Tyagi. Next, he did a postdoctoral fellowship at the Institut Pasteur in Paris as a US National Science Foundation Fellow. He worked on gene expression and nuclear organization with a focus imaging of RNA and transcription. Next, he started his own lab and moved to South Africa a little over 10 years ago. Recently, he moved to the Radboud University in Nijmegen in Netherlands, where he chairs the Department of Cell Biology of the Faculty of Science. He's also a member of the Department of Human Genetics at the Radboud University Medical Center. Musa, welcome. Thank you very much for your participation.
Thank you very much for inviting me, and it's really a pleasure to be participating in this HCA workshop. I am always very happy to see how the HCA community is expanding in Latin America. I'm just going to share my screen. I hope this works. Can you see that? I guess people can see that, so
yes,
it should be fine.
It's working for me.
So today I'm going to talk a little bit about two different projects we have in the lab. One project is, they're both related to spatial transcriptomics. One project is subcellular mapping at single cell resolution of RNA and protein. And then the second project is the equivalent of doing single cell RNA-seq in single cells using spatial transcriptomics and looking at dozens of genes in each cell with subcellular resolution using some new microscopy tools that, that we've been able to collaborate with an industrial partner on. So we all are very excited about the prospect of spatially resolved transcriptomics, and that's essentially because it introduces the possibility to use non-biochemical approaches to be able to really know the spatial distribution of where cells are, and also to be able to identify where gene expression in spatial position is variable. And that's been a long-standing goal in biology. And one of the key challenges has been, how do we see many genes or RNAs and yet have such a constrained number of fluorescent tags to be able to see them? And so we have many ways that have been developed to visualize RNA. One of these ways has been using a technique called single molecule FISH, which came from the lab where I did my PhD in New York. And this basically involves using very short probes, like you can see here, which are tagged with a fluorescent dye. And that fluorescent dye allows us to be able to decorate an RNA transcript in one of a preset number of fluorescent colors. And usually, just because of the spectral landscape of these colors, we can only do this reasonably in 4 colors. And then the lab of Xiaowei Zhuang developed MERFISH, which is basically a sequential barcoding approach where we can expand the number of transcripts that we can see at any one time by essentially delivering a set of probes and then removing or extinguishing the fluorescence from those probes, and then delivering a new set of probes. It could even be in the same color because you've extinguished the previous set of probes. And this allows you to do repeated staining and imaging and extinguishing, and then registering the position of each of these different RNAs that you've decorated, and continuing and resuming that process again several times, usually until the sample itself is completely degraded. And that technique is most, I think its best approach is called MERFISH, which Jeff Moffett and Xiaowei Zhang have really expanded dramatically on. But there are other techniques that involve spatial barcoding and spectral barcoding. But I'll start off really by helping us understand why we're interested in the first problem I talked to you about, which is this idea of understanding the subcellular localization of RNA. And we know in eukaryotic cells that there's a spatial separation of transcription and translation, where you have DNA being transcribed into RNA in the nucleus, and the conversion of RNA to protein occurring in the cytoplasm. What's far more intriguing is that this conversion of RNA to protein in the cytoplasm seems to involve the localization of many different types of transcripts to different subcellular locations, depending on the functions of the proteins that will emerge from those RNAs. And this is really espoused in these different models that I'm showing you here. One of them is the enterocytes of mice, where you have grandiole-dependent apical localization of ApoB to the apical side of the enterocyte. And in another model here, in neurons, where you have the localization of specific RNAs to specific positions along these dendrites in these neurons, depending on signals that are being transmitted in the brain. So these are just examples of where there is not only the translation of RNA to protein in the cytoplasm, but specific positioning and localization of RNA to locations that are important for the functions of the proteins that emanate from those RNAs. And really, a big challenge is that as much as we can develop lots of gene expression techniques to be able to understand gene expression and the levels of genes that are expressed, and these are all biochemical techniques, how do we recover where these RNAs reside at subcellular locations within cells, which can inform us more deeply of their functions, like you can see in these examples here. And so for cell biologists, the main challenge has been to take these sort of in vivo whole organism models and translate them into cell biology models that we can grow in culture. And one way to do this has been to really use techniques where we can reconstitute cellular microenvironments. And the way we do that is by constraining the geometry and architecture that is present in vivo, but redoing that in sort of in vitro settings. And so you can see here in this review that's several years old now, where you can sort of see that cells growing in different organisms and tissues, depending on the tissue, have really constrained geometries and architectures. And these geometries and architectures impose specific subcellular behaviors on the proteins and RNAs that are distributed within these cells. And so Manuel Thierry really has been one of the leaders in this field. And one of the key developments was the ability to micropattern cells and to essentially show that constraining the geometry and architecture of cells leads to highly reproducible internal organization. And that highly reproducible internal organization results in these beautiful organizations we see of cellular structures on micropatterns such as actin and microtubules. And the way you do that is really to alter the adherence of the surface of glass, and that forces cells to grow in specific configurations and where they assume these beautiful micropatterns. And when you take detailed subcellular measurements of these micropatterns, of cells on these micropatterns, what you see is that proteins start to adopt reproducible subcellular localizations that can be measured with very high fidelity, showing us that indeed the cells seem to have specific, if you like, GPS coordinates for the vast majority of proteins that are made to direct them to specific locations within the subcellular volume. But the real question is, do these same GPS coordinates also exist for RNA? And if they do, maybe micropatterns could be a way for us to be able to reveal that, at least technically. And so in the same way we can generate tSRE and UMAP plots, we can see that maybe by taking tissues and understanding their localization of RNA within these tissues, we may see that even a specific cell type that we determine by single cell RNA sequencing with a copy number of a specific group of transcripts that are denoting that this cell type is this purple cell type here, the copy number may be the same, but actually, if we were going to look inside that specific group of cells that are in this tSRE, we may find that actually that RNA is similar in copy number across these three cell types, but actually the RNA is localized at very different locations and those three different cell types, suggesting that there's this cell, even though it has the same number of copies of RNA, is actually in three different states because the RNA is localized to three different locations, resulting in essentially proteins being at different levels of activity. And you can imagine this can be quite confounding in many situations where we could believe that cells are actually expressing a gene that's being made at a protein, and that protein is actually equally active in these different cells because they occupy the same tSRE or UMAP space. So this was really what motivated us to develop techniques to be able to better interrogate this relationship between RNA distribution, RNA copy number, and proteins being made. And this is really like one of the deep motivations of us pursuing this approach. And so single molecule FISH presents itself as a very good way of doing this in combination with micropatterns, as I've described. And here we're just showing an example of how the combination of the use of micropatterns in single molecule FISH can enable us to decorate specific messenger RNA called ARDIA. And this localization pattern can be further decomposed using different techniques, such as denoising and aligning multiple cells that grow in micropatterns to see the reproducibility of this RNA spots that we see over dozens and dozens of cells. And so we can then overlay that distribution analysis with the detection of specific organelles and structures within cells. And one of those structures that's also highly reproducible within cells is the MTOC, the microtubule organizing complex, which always seems to localize to one position, the central position within the subcellular locations within cells. So when we take measurements in this sort of RNA distribution analysis over hundreds of cells, what we start recovering is that the number of copies of RNA we detect and the number of and the size of the cell are highly correlated with each other. And that's quite different from what we see in standard cell culture, where we can see variable numbers of cells, where actually the cell size and the cell arrangements seem to obscure consistency in the number of transcripts that are detected. So you can see that here on the left, we were detecting this by single molecule FISH in standard cell culture. And on the right, where we do the same experiment, where we're using tuned sizes of micropatterns, you can see a much tighter correlation between the number of transcripts and a specific gene and cell size. So we can then use different types of analytical approaches to see where different RNAs are localized in the entire cellular volume. And for example, RNAs that are localized very close to the periphery of cells, say when they receive a polarization stimulus, can be mapped using these different techniques where we use the peripheral fractions and increasingly small fractions that go towards the periphery, and determining the concentration of at single RNA resolution of where these RNAs are localized. But using these techniques, at least from a mathematical point of view, are not so robust. And so to gain more confidence in our approaches, we decided to use something called a Ripley K clustering approach. And this allowed us to fit the distribution of RNAs at the subcellular level into two models. One, which is complete spatial randomness, where essentially RNAs are randomly located within the cellular volume, or another one, which is basically spatially clustered, where we can see enrichments of RNA in specific subcellular locales. And enrichment of RNAs would, for example, correspond to some of the early images I showed you, where in the enterocytes, you have accumulations of a specific RNA species at the apical part of the enterocyte. So by applying a Ripley K algorithm to RNA data sets such as these, where we're counting individual molecules and able to recover their spatial distribution, we really can assign a clustering index, which allows us to know when a transcript is either spatially clustered or really corresponds to a model of complete spatial randomness. And so when we do that with a set of RNAs that I just showed you earlier, which have different propensities to accumulate at the peripheral edge of a cell that is polarized, what we see is that depending on the transcript, we have different rates at which the clustering can accumulate at the periphery. And here in the solid lines, we have the clustering of the RNA. And then temporally, we can see the clustering of the protein, which should occur later than the RNA. And that's telling us that, for example, here, beta-actin first accumulates into a cluster. It doesn't tell us where it accumulates, just that it is following a spatially clustered model in the Ripley-K algorithm. And then later on, the protein is also clustered in that region. And as I mentioned earlier, this follows sort of the basic dogma of information that the RNA is first made, and the RNA is made into protein. And if the RNA clusters in one location, then presumably the protein will cluster in a similar location if that's part intrinsic to the function of that RNA. Contrastingly, an RNA like GAPDH does not follow a spatially clustered model and follows a more complete spatial randomness model. And there we see that indeed we have far less clustering of GAPDH protein, and we see that this follows a far more random distribution of the protein, indicating that for GAPDH function, subcellular localization of GAPDH is less important. And so we could reliably develop this sort of clustering to determine non-random versus random clustering for us to make our algorithm far more robust in terms of being able to sort of denote whether a gene or an RNA can follow these two different models. We then could superimpose the role of organelles within cells, and one of those organelles is the microtubule organizing complex, and that's able to really allow us to more tightly link the cell's understanding of its central position, which the MTOC always marks, and the relationship to where RNA localizes vis-a-vis the MTOC. So you can think of the MTOC as a kind of guide that tells a cell this is the absolute center of the cell. And then you can imagine that it can dictate whether an RNA localizes at different hands of the clock from 1 o'clock all the way to 12 o'clock. And here we've just partitioned those different positions into kind of 4 different quadrants. And here you can have RNAs that are following the sort of 12 o'clock model of where the MTOC is located. And in this second model here, RNAs follow the 3 o'clock model, or the MTOC can reposition the center of the cell to 3 o'clock, and the RNAs go to the 12 o'clock position, and so forth and so forth. And this allows us to derive something you can call a cytoplasmic mitochondrial polarity index, and that allows us to relate the MTOC's position in that index directly to where the RNA is localized. And if here, for example, we can see after several hours, the ARCDIA RNA is tightly related to the 12 o'clock position of where the MTOC is, indicating that polarity is highly deterministic of where this particular messenger RNA will localize. And we can see that happens to go a little bit different for this particular RNA, where at first polarity is highly dictating its location, but there's a dramatic fall over time as if the loosening of polarity and the localization of part D3 are occurring over time. So as I've mentioned to you before, these are cluster measurements, and we have to kind of relate them to actual spaces within cells so we can subdivide areas of the cell. And by subdividing these areas, we can assess RNA and protein in these areas where we can see that the sublocalization of an RNA also tightly relates to where the actual protein is made over time. And this is important because it allows us, for example, to look at the GAPDH example I showed you earlier and see that we have indeed over time the accumulation of a random distribution of the RNA, you can see here in red dots, and a random distribution of the protein, suggesting that the RNA and protein localizations are not tightly linked. And we can even, as we have a single molecule resolution over the RNA, we can look at copy number of the RNA over time. And we can contrast that with, say, beta-actin, where the relationship between the RNA and the protein is much tighter, and wherever the RNA goes, the protein also is made at that same location. So once we've developed these mathematical models, we can obviously apply these to different situations. And one of these that we applied them to is sort of muscle development, one of a long-standing question, because muscles are multi-nucleated. And when we do that, we can see that there is a very tight relationship, and I'll just summarize the data very quickly here, but there's a very tight relationship between a protein structure that occurs periodically between two nuclei in muscle cells and the accumulation over time to an RNA that is developmentally localized to exactly these same sorts of striated lines. And so to do that, we actually analyzed the messenger RNA accumulation using the algorithms that I've just mentioned to you so far in this talk in mouse developing muscles. And there we could see that as the RNA clustered over time, the RNA appeared to be randomly distributed in time. And we could see the emergence of the Z-line protein starting to nucleate at different positions in the periodic manner I showed you earlier. And the particular RNA in question is actin-2 messenger RNA. And over time, as we followed the actin-2 messenger RNA subcellular position between these two nuclei within the muscle fiber, we could see that the Z lines at the protein level started to form along the accumulated areas at a certain concentration of a certain number of messenger RNAs as they accumulated in striations. And so we could also compare this, say, to GAPDH, which I told you earlier sort of randomly distributes, and GAPDH doesn't seem to accumulate in a similar pattern with these striations, suggesting that its localization isn't deterministic or correlated with the Z-line formation. So this sort of reveals like state changes in the way that these striations form. And we know that essentially the copy number of messenger RNAs, if you were to sequence this by single cell RNA sequencing, isn't very different between the immature and mature muscle fiber, but yet the actin-2 messenger RNA positioning is sort of determining where we get the position of the striated protein. And so if you want to like overview the techniques that we kind of have developed so far, they really involve the ability to do this systematic image acquisition that allows us to recover 3D cellular information and relate that to RNA and protein distribution. And once we do that, we are able to see different subcellular calculations of RNA protein spread, how RNA protein clusters accumulate over time and relate them to specific organelles such as the MTOC. And then we are able to apply this analytical framework to multiple types of cellular models, from essentially tissues like muscles to constrained models like micropattern cells to unconstrained models like cells growing in culture. So this is all really nice, and it gives us a whole basket of tools. And you know, this work kind of predates a lot of the technologies that I'm going to show you in a moment that really allow us to kind of scale the data that we're able to collect, not only from a single RNA, but potentially from dozens, if not hundreds of genes in a single cell at a time, and be able to uncover highly interesting models of how not only what a cell state is, but also relate the subcellular positions of RNA and proteins simultaneously in one cell. And so our journey on that begins with another group in our lab, which works on microscopy. And we've been working a long time with a company in California called Optical Biosystems, who had developed a technique called synthetic aperture optics. And synthetic aperture optics really allows us to do something that is sort of violating Snell's law, which is we're able to have a very large field of view and use a very long working distance lens like a 20x lens, and yet be able to have a very, very, very high magnification. What does that mean exactly? Well, what it means is that here's a field of cells where we can acquire hundreds of cells that have been stained with the gene here, TFRC. And this is one field of view where there are, literally hundreds of cells that have been stained with single molecule fish. And you can see the DAPI here, and we can go and examine at single cell resolution and count the number of RNA copies offline after acquiring this at a 20x resolution. Now, this technology at the time, in this iteration on this instrument, could only be done for like one transcript at a time. And what's emerged recently from optical biosystems, and renamed sort of Rebus Bio, is the ability to do the same kind of acquisition over dozens of genes. But they're not the only ones who can do this sort of subcellular spatial transcriptomics. Recently, another instrument called the MERScope was released by a company called VizGen, and this uses the MERFISH protocol that I showed you very early on in the talk to be able to acquire subcellular RNA fish information from hundreds of genes using the MERFISH protocol. So on the left, we have the VizGen from VizGen instrument, the MERScope, and on the right, we have the ESPER from Rebus Bio, which is the instrument that we have in our laboratory. And so how does this work? Well, essentially, this instrument can do single cells, but it also can do something even more exciting, is that it can do tissues, and we can basically use cryosection tissues that are fixed on conventional cover slips. We load these into a flow cell, which has got a microfluidic architecture, and then we can start a run on the instrument, where in this run, we can flow a whole set of different single molecule fish probes, essentially using the set of protocols that I showed you earlier. But I'll show you that again so you can sort of understand how that works. So to just reprise what I mentioned to you before, this allows us to essentially, in the microfluidic chamber, stain with a set of probes, image where those probes are, then change our laser to extinguish those probes by cleaving the dye from those probes, and then use our microfluidic architecture to wash the flow cell and introduce a new probe. And at the same time, of course, we can register and capture images of the location of each of these prior events. And that allows us to continuously probe over time. And this process takes literally 24 to 36 hours, but to continually process over time inside a tissue. And so if you want to look at that process here, this is reproduced from a paper from Xiaowei Zhuang's lab in the early days of MERFISH, where you can hybridize with single molecule fish, bleach by here in this case, by using a laser that bleaches out that dye, and then by microfluidics introduce a second hybridization event. And in this case, they could do up to 16 cycles of hybridization. And each one of these cycles is sort of capturing a different transcript, and those different transcripts are located at different locations within these, in this case, these were cells. And so you can imagine here, at 16 cycles, at least using MERFISH, where they're using a Hamming code, you could probably detect up to 50 to 100 genes. In our case, and this is sort of test data that we've been using now, we stitched together an image using synthetic aperture optics of multiple sections here of our test tissue. In our case, this has been a synovial joint tissue, which we are looking at 30 different genes stained over the entire synovial joint using spatially resolved transcriptomics, so 30 different probes to 30 different immune acting genes. And we have two types of synovial joint, I'll tell you now, which are hyperinflamed joint and a joint where we've used a particular drug to be able to attenuate hyperinflammation. And we want to see the spatial organization of where different cytokines are being expressed. And I'm sorry, we haven't really used CellDex here to be able to map the reference genes, but we have mapped the genes themselves to a UMAP, as you'll see in a moment. So I'm going to zoom in this area, and what's really exciting about this is that, as I showed you earlier, we could do that with cells, but now we're doing that with tissues, and you can see behind where around these nuclei from the segmentation, you can see the DAPI staining in gray, and you can see the multiple colors where each of these 30 different genes at the level of single molecule fish has been staining, and we can actually recover the subcellular localizations of each of these genes. And so here, just because the size of these files is enormous, I've tried to do as much compression as possible just for the video, but we're talking about file sizes that are well into the hundreds of megabytes to be able to visualize them. And what we can then do is see this from a different point of view, where we have different cycles of hybridization. And this is over the same tissue, where we can see the entire tissue looks like this, but if gene 1 is only staining in this area, gene 2 stains in this area and continues to stain different areas, and gene 3 stains more at this distal location in this. And we can do this over and over again in this cyclic hybridization pattern to achieve all the way to 30 different genes. And once we have those 30 different genes, we can convert this data into similar data that we would have from single cell RNA sequencing data, but these matrices in these tables are just being recovered from counting spots and segmenting them into cells that are made from single molecule RNA fish. And so we can use all overlap of a lot of pipelines that we normally use from single cell RNA sequencing data. And basically this is just one of them, which shows the dispersion of highly variable genes in terms of immune expression. We can generate similar UMAPs. Here in these UMAPs, we are able to score 7 different types of cell types in these different dimensions. And we'll go into this data a little bit, but as we go through these UMAPs, we can see that over the different cycles of these genes, we can see where each of these genes that I show you, for example, gene 1 is represented in over all these parts of the UMAP. And as we progress, gene 2 is equally represented in some parts of this, and over and over and over again as we cycle up to 30 genes. And I'm showing 19 here. We can rebuild our UMAP, but we can rebuild it in 2 ways. So here we're building it sort of in a graph format over 2 axes, but I'll show you later how we can rebuild the same information spatially over the entire section of the CRES section that we've done this. So again, there's a high degree of overlap on these pipelines, where for each of the different cell types, we can relate the expression of our group of different genes here in UMAP space. We can also relate the gene expression level for each cell type, where we have a predominant type of gene. For example, gene 1 is really the predominant gene that's expressed in cell type 7, which in our UMAP space is over here. And you can see the degree to which this is expressed. And we can generate volcano plots. We can generate plots in terms of the counts. This is the actual number of single molecule fish detections that we have across each of these genes. And as I mentioned, we can also generate an entire map, an entire spatial map, where we have basically the same information that we have in the UMAP, but superimposed on the section, on the CRES section itself, where each of these dots represents a cell, where we have a specific population of these 30 genes, when we can call the cell types based on these 30 genes that we're detecting in our experiment. And so today we are at around 30 genes for this. We are already developing different approaches in the lab that'll allow us to increase this number tenfold to up to 300 genes over 30 cycles of hybridization using techniques that are not too different from the techniques of Murfish. But this really brings us to a really exciting era where there's going to be a very high convergence between data that we get from biochemical techniques, where we really disintegrate cells and count the number of molecules, to spatial transcriptomic techniques, where we actually leave cells intact within tissues and count the number of RNA molecules that we get in situ. And really, I see this as just the beginning, because the other project we have in our lab is to superimpose the RNA fish data with protein expression data that we will also use cyclical hybridization approaches and antibodies like codex to be able to detect. So with that, I'd like to just end my talk by thanking all the people who have helped us in this project. And I'm sure they'll come up in a second. Yep. So this project was really at first driven by a staff scientist in the lab, Anna Savulescu, and she worked with Robin Bracken, who's now at the Leibniz Institute in Germany, with Jan Schmorenzer. We collaborated on a lot of the bioinformatic pipelines and the development of a lot of the methods that in the first part of the talk with the Nikolsky lab, and especially with Emmanuel Bouillol, who's a PhD student in that lab. We also developed a lot of automation for this with Stéphane Dagonville, and the mouse model that I showed you came from the lab of Edgar Gomez. The tissue data that I showed you was from Ezio Falk and Stephanie Fanucchi, and we collaborated on that with Rebus Bio, which is one of our first samples of doing that. And of course, we're moving on to other things. Our work is supported by the Chan Zuckerberg Initiative, and also by other funding from the Radboud UMC and the Radboud University. So with that, I'd like to thank you for your attention. I'm very happy to take any questions you may have. And thank you so much for inviting me.
Thank you very much, Musa, for your wonderful talk. We have some questions from the audience. Let me just check here. The first one from Laura Gomez Romero. Great presentation. During the processing of these huge imaging files, how accurate is the segmentation process? Does it take into account that a tissue is composed by different cell types and each type could have a different form?
Yeah, this is a really great question. Segmentation is one of the biggest, cell segmentation is one of the biggest challenges that we have in doing this. I kind of did a little bit of a sleight of hand here, but the audience is very smart, like people like Laura, extremely smart. I didn't show you the data sets where we have specific stains that allow us to stain the sort of cellular membrane. And by being able to do that, it solves a lot of the image processing for the cell segmentation to occur. So that sort of sidesteps that question. It is not an easy thing to do that type of cell segmentation offline, but you know we have a number of tools that enable us to do that, and of course we're using a lot of like complex image analysis to be able to detect these different subcellular boundaries between cells.
Great. Now I have a question from Sebastian Urquiza. Good presentation and very interesting. I would like to ask you about the polarization of the transcripts in the cell, because the cellular system is so dynamic. It will be possible to detect across of cellular biomarkers the polarity of the transcripts and make the analysis more specific?
Yeah, so I mean, this polarity question is actually a question we got from reviewers. It's a very good question. I think we are very wary that not all cells polarize in the same way. And as you say, you know, these cellular systems are very dynamic. So detecting this across multiple cell types for us is something that we haven't done, but we are hoping to do now with our spatial transcriptomics platform on tissues. And the way we want to do that is we want to look at polarized epithelia and label the MTOC, which we think is going to be a very good marker of polarization. And as you say, of course it's very dynamic, and there may be more disorganized tissues, non-epithelial tissues, where cells don't have the same architecture, where we can compare the position of the MTOC. So I don't know if that answers your question. It's been long-winded way of answering.
Thank you, Musa. Another question is from Tiago Lubiana Alves. Thank you for the beautiful talk. Amazing images. It would be beautiful to see the RNA movement too. Is there any way to look at RNA molecules in living active cells? Something analogous to GFP tags used for proteins, for example.
Well, Tiago, you're in luck. I'll share my screen if you want. I actually skipped that slide, but just for you, I'm going to show that. So here we go. So this is work that I did like over 11 years ago, where we used something called a molecular beacon to be able to decorate this RNA you see in red here as it moves within this Drosophila oocyte. And so it is possible to do this. It is a very difficult experiment to do to maintain these cells alive and to be able to sort of inject the probes or deliver the probes inside the nuclei of these cells. So we deliver them into the nuclei of this oocyte. And then we could actually track these in real time. So this is one of the technologies out there. There are some other technologies like MS2, but that involves like a lot of genetic engineering of the transcript. So yes, but great question.
Amazing. The next question is from an anonymous attendee. What explanation can be given to the difference in the location of an mRNA between cells whose expression pattern is shared?
Yes. This question for us is really a question of cell state. So that's the explanation we would give. And I tried to kind of conceptually show that a cell can be expressing the same transcript as its neighboring cell, but that transcript can be in a completely different location, subcellular location, because of the sort of inchoate state of the cell. And we're seeing that a lot in macrophages, for example, where macrophages can pre-position RNA already made in different subcellular locations where it can be translated very quickly. And so that kind of tells us a little bit about that particular macrophage or monocyte, where the same transcript won't be sort of localized in those areas, but you know, those two cells are the same cell type.
Another anonymous attendee, great talk. Could you say more, some word more about the technical needs for these kinds of RNA cell visualization in terms of microscopy?
Yeah, so we're very fortunate. I mean, you could set up a similar system as the VisGen, let's say, because the VisGen is using conventional optics. So that is not the system that we have. The VisGen has the advantage that it can, you know, using wide field fluorescent optics, very well tuned lasers, you can, if you have your own microfluidic system, you could build something like the VisGen. So Jeff Moffett has done that at Boston Children's Hospital. The VisGen instrument is, you know, at a great price as well. The nice thing about the Rebus instrument, though, is that it has a completely different set of optics, a synthetic aperture optics, slightly more expensive than the VisGen, but it does this amazing thing of, like I showed you, where you can zoom in from a very large image into a very narrow space, and at subcellular resolution captures such a high degree of information. So I think those are the basic approaches, the two approaches to get subcellular resolution. 10x does have a solution called a Visium, but that isn't subcellular. That's its resolution is very far from subcellular. So you won't be able to capture the kind of information that I've been talking about here. But yeah, so that's what I would suggest.
Perfect. Another question from Marco Antonio Fonseca, and it's a question that I also would like to do. Great presentation. Have you tried to analyze the spatial transcriptomics of long non-coding RNAs?
Yeah, I mean, our lab works very heavily in this area. And so, yes, we're definitely doing that. Yep, definitely.
Amazing. Another anonymous attendee. In general, there are many single-cell RNA-seq analyses without integration of spatial transcriptomics. What do you think are the pros and cons of this?
Yeah, I mean, there's some very exciting techniques, like from Nicholas Rajowski. I was going to talk about that, but I didn't have time. He published a very nice paper in Nature a couple of years ago, where he kind of compared single-cell RNA-seq data and spatial transcriptomics data and showed how you could integrate these two data types. I would encourage you to go in the literature and look for that. And please email me and I can send you the paper if you can't find it. But I'm very happy to point you in that direction. I think that type of integration is going to be routine in the future because we are already working with this microscope with tissues from patients, arterial plaques, as I said, synovial joints, human fetal brains. We are doing a lot of techniques in the clinic where we can actually do single-cell RNA sequencing and spatial transcriptomics from the same sample.
Thank you. And now I have a question from Carla Vargas. Thanks for the presentation. On a sample processing level, 24 hours analyzing the same tissue looks like a very long time. How the method could affect the outcome? Would the method affect the outcome?
Yeah, so, this long-term acquisition by cyclic hybridization, it's constrained by the imaging time. And, I think you do make a good point that we can only do that many cycles because literally from exposure to the light and the lasers and all the hybridization events literally destroys the tissue. I really believe that we're getting closer to a ground truth, but I also would not dismiss your criticism that these repeated cycles, which are increasingly destructive, could be affecting the readout in terms of the copy number of RNA. Now, bear in mind, we do a lot of chemical fixation to protect these RNAs from degradation. And we don't have evidence that repeated exposures to light degrade RNA in this way. But yeah, I mean, it's definitely possible. I think there's another question.
Yes, there is another question from an anonymous attendee. Great presentation. Regarding the mRNA clusters, have you had the possibility to verify if they correspond to ribonucleoprotein complex, such as P-bodies?
Yeah, so that study that we did, it's on the bioRxiv, so you can go take a look. But none of the genes that we looked at, the RNAs that we looked at, were RNAs where we could tell in the literature that they were associated with RNPs. you could say that beta-actin in the cell type, because beta-actin has been shown, for example, by the group of Sanjay Tyagi in neurons to be associated with RNPs. But P bodies is a specific type of RNP. It's kind of a very, very specific type of RNP, and not from the genes that we've looked at. It would not surprise me from other genes that they were associated with P bodies.
Perfect, Dr. Musa. Thank you very much for your participation.
Thank you so much. It's really been a pleasure. And you know, next time we'll have more data sets, and hopefully we'll be able to give some to your students in the workshop to help us looking for all kinds of data scientists. So if you're looking to do data science and this kind of thing, you know, please come and join us. Have we integrated?
Yeah, there is another question here with at the chat. Let me try to find it here because it's not in the.
Yeah. Have we integrated multicolor microscope? Yes. So our microscope is multicolored. It can do 4 colors. It has multiple lasers. I didn't really go into that technology. So yes. However, you know, you can do this with very few colors, like 2 or 3 colors. But yes, so it is multicolored. But the cyclical fish helps us to get around this problem.
Perfect. Thank you, Musa.
Thank you. Really, thank you for inviting me, and I'm always happy. You know, as soon as this pandemic ends, I think one of the first places I'm going to go is visit you guys in Brazil. So thank you so much. Really appreciate it, and keep the HCA going strong over there.
It will be amazing. Also, it will be amazing to have access to the data sets that you mentioned for the hands-on training. And of course, we're planning to do these in Mexico, in Chile, and in Brazil, and other Latin American countries. The idea is to spread the word on this technology here and build capacity locally. Thank you.
Awesome. Thank you so much. Thank you. Goodbye.
Bye-bye. Now we move on to our next speaker. Dr. Mena Cletworthy is the professor of translational immunology at the University of Cambridge, Department of Medicine. She also works clinically as an honorary consultant nephrologist and holds an associate faculty position in cellular genetics at the Wellcome Sanger Institute. Her research is focused on immune regulation, including the role of tissue environment in shaping resident immune cell phenotype, function, and interactions, particularly in the kidney. To follow the study of human cells, the lab has established permissions and protocols for isolating and studying immune cell subsets from human organs. Dr. Mena, thank you very much for being here. Welcome to our workshop.
Thanks very much. Thanks very much for the invitation. Are you able to hear me and see my screen okay?
Yes, it's working perfectly.
Great. So as you've said, I'm going to talk to you about our efforts to make a human kidney cell atlas, and also give you an idea of how we might use it. Now, I thought it'd be useful to just briefly tell you about my perspective or my background, so how I come to this. So I am a clinician scientist, okay, so I work as a kidney doctor, and mainly what I do is look after people with autoimmune kidney diseases or those who've had kidney transplants. And so I guess that the work of my laboratory is grounded in trying to base our observations on what we find in human tissues. And then we also have a sort of more basic science, cellular immunology component within the MRC laboratory of molecular biology in Cambridge, where I'm based. And then I have, as you've said, an associate faculty position at the Wellcome Sanger Institute, and I'm based in cellular genetics, which is where Sarah Teichmann is also based and leads the cellular genetics team. So in that aspect, we very heavily rely on single cell technologies. And so I'm interested in tissue immunity, and we've got a few organs that we focus on, but today I'm going to tell you about our efforts to generate a human kidney cell atlas. So the outline of what I'm going to talk about over the next, say, 45 minutes are basically to give you an introduction to the kidney and to talk about why we might need a kidney cell atlas. And then to tell you about how we have gone about making version 1 of the kidney cell atlas and some of the challenges there. And then finally talk about how we might use the kidney cell atlas, so particularly from a translational and medical point of view. So the kidneys, I'm going to start sort of from a very basic level, assuming that there may be some people that are completely computational, don't know much about biology. So just so we start from the same place. So we have 2 kidneys, or most people have 2 kidneys. They're about 11, 12 centimeters in length, and they sit at the back of your abdomen. And their job is to remove waste products and acid, to control water balance, and thereby they control your blood pressure. The kidneys also make a hormone called erythropoietin, or EPO for short, and that stimulates your bone marrow to make red blood cells. So the kidneys are critical to stop you from becoming anemic. They also make the active form of vitamin D, so again, help to balance your calcium and phosphate metabolism. So without them, you get bone problems. So they're essential for life. If your kidneys stop working, unless you have dialysis, you'll die because of the buildup of waste products. And so anatomically, the kidneys are made of an outer cortex, an inner medulla, the pelvis and ureter. So blood is filtered in units within the cortex, which I'll come to. The filtrate is concentrated in the medulla and then flows on into the pelvis, through the ureter and out into the bladder. And the individual subunits of the kidney are called nephrons. And each nephron is made up of a glomerulus and ultrastructurally. So if I tell you something about the cells, because I'm going to come back to this. So blood comes in through this blood vessel and is then squeezed through the capillary loops. So there's specialized blood vessels here. And then these blood vessels are supported by epithelial cells called podocytes. And these are really unusual. They're large and they have these foot processes which engulf the glomerular capillaries. So then urine or the filtrate is squeezed out into this space and then flows through the proximal tubular cells, where a lot of the useful things that are squeezed out are reabsorbed. So those cells are very metabolically active. Into this bit here, which is called the loop of Henle, and this enables you to generate a sodium gradient in the kidney, which ultimately allows you to concentrate urine. Distal tubule, and then the collecting duct and out into the outside world. And as well as these components of the nephron, which include the epithelial cells that are all along here, endothelial cells, and the mesangium that supports these capillary loops, we also have known for a while that there are resident immune cells, so of particular interest to me. And really the best described of those, and mostly, or most of what we know about them are actually from mouse models, are cells called mononuclear phagocytes. And there are 2 flavors of these, macrophages and dendritic cells. But in both cases, they're able to engulf things, that could be bacteria or it could be cellular debris. And in the case of macrophages, they get rid of it. In the case of dendritic cells, they actually process that information and use it to activate the adaptive arm of the immune system. And we know that these resident kidney cells, and again, this is mostly based from mouse work, completely based on mouse work, can either get there from circulating precursors, so monocytes, and then they come into the kidney and become macrophages, or actually, there's a subset that are prenatally seeded during embryogenesis, and we think that these cells may have different functionality. So you might say, well, why are there immune cells within an organ like the kidney, given what you've said about its function? Well, actually, the kidney experiences a number of different immunological challenges. So most obviously, is infection. So urinary tract infection is common. So particularly lower urinary tract infection in the bladder affects more than half of women in their lifetime and about 10% of men. And some of those bacteria are able to ascend up the ureter and into the pelvis and infect the cells in the middle bit of the kidney here. But then conversely, from the circulation, sometimes there are autoantibodies that are able to recognize components within the glomerulus and can cause inflammation and damage. And that can happen in diseases like systemic lupus erythematosus or glomerulonephritidis. So you can see that, I guess, to summarize, that the immunological challenges that affect the kidney come via very distinct anatomical regions. So one from the bottom and the other kind of from the top. So why does all that matter in terms of immune responses in the kidney? Well, kidney dysfunction is actually phenomenally common. So there are around 600 million people worldwide that have chronic kidney disease. And if it reaches end stage, of course, it means you either need dialysis or a transplant. And if you can't access those two things, then, unfortunately, ultimately people die. So the chronic kidney disease is characterized, if you look under the microscope, by the presence of scarring. And it can occur due to a whole variety of things. So ischemia reperfusion injury. So that is if you get a low blood pressure or you're ill and septic. So for example, some patients with COVID-19 would get kidney injury related to that. Then there's glomerulonephritis, which, as I've described, can happen if there are autoantibodies that bind. There are common things like diabetes and high blood pressure, and those can lead to damage to the kidneys or ascending infection. But regardless of the insult, actually, we know, or at least from mouse models, that the resident immune cells, so particularly the mononuclear phagocytes, determine the response to these different perturbations and ultimately will determine whether you resolve them or whether you progress to fibrosis and chronic kidney disease. So I suppose, to summarize, why do we need a kidney cell atlas? Well, we need it to better understand at a much higher resolution which kidney resident immune cells are present in the kidney and what immune capability do they have. Functionally, what is it that they do? And obviously their transcriptome can give us an idea of that. And before we move to disease, it's really important to know what the ground truth in homeostasis is, so we can understand what goes wrong and also can better think about how to make it better, so to identify molecular targets for disease. So what do we do currently? Well, actually, the clinical situation is still relatively rudimentary. So the way in which we make a diagnosis of a kidney disease firmly is to do a kidney biopsy. That's done under a local anesthetic. We get a small piece of tissue, and what we principally do is look at it under a light microscope. And we're able to make a diagnosis based on the appearance of different cell types. So for example, here, these round structures are kidney tubules. And if there's the presence of immune cells, so some of these darker nuclei, then it might tell us, for example, in a transplant, there's rejection, in a native kidney that there's glomerulonephritis. We also have gone to the second level of we do routinely try and stain for some proteins. So for example, this shows staining for IgG, so one of the antibodies, so that can tell us that there may be IgG deposition. And more recently, we've actually progressed to, in some contexts, so particularly in transplantation, using molecular pathology, so where we do a bulk measurement of mRNA. And so, for example, in transplantation, we now know that the gene sets that are associated with T-cell mediated rejection versus antibody mediated rejection. But of course, there are limits to this because although we might say that we appreciate that some of these transcripts are characteristic of, for example, endothelial cells or T cells, bulk RNA sequencing doesn't allow us to specifically associate a transcript with a cell type. So to summarize this first part, the kidneys are really important for homeostasis. If they don't work, it's a problem, And if they're completely dysfunctional, without treatment, you'll die. Chronic kidney disease is common and it can be devastating. Immune responses contribute to kidney pathology and are important determinants of outcome, so resolution versus scarring. And our conventional approaches to understanding the molecular contributors to kidney disease pathogenesis and progression are pretty rudimentary. So we use cell morphology based on microscopy, detection of a limited number of proteins like IgG or complement components. And at the moment, in a more limited way, we also apply molecular pathology using microarray or bulk RNA sequencing. But that still isn't really routinely available clinically. So that gives you an idea of, what the need is and why we might need to make a kidney cell atlas. So I'll next tell you sort of how we went on to do that. So I guess the first thing that you need is a tissue source. And I think, for many of the atlases, this can be a major challenge. And our options range from tissue biopsies or organ resections. And these samples are extremely fresh because we typically get them from the operating room. But one of the problems for biopsies, they're often very small because the person still needs that organ. If you're taking an organ out, and we do take kidneys that have been resected, that's often because they contain a cancer. And so you need to find an adjacent normal bit. And even then there's some question about how normal that is. Then another potential source that we've used a lot of are organ donors. So these are individuals who've consented to have their organs used for transplantation if they're suitable. And some of those also consent for research in the UK. And so those individuals were able to take samples of other organs. And in the UK and in Cambridge in particular, we have something called CBTM, the Cambridge Biorepository for Translational Medicine. And this provides an infrastructure for sampling organs from donors that consent for research. And this is also relatively fresh, although, obviously by the time individuals become donors, they are deceased, but these samples are being immediately taken. And then at the other end, there are post-mortem examinations, and for example, as undertaken by GTEx. I mean, one issue with that, it depends how rapid they are, and obviously RNA can degrade pretty quickly. And then the other considerations are around data consent. If you've got someone who's deceased, that might be easier than from individuals who are still living. And also access to tissues. So biopsies, you're very limited in what you can get from one individual. It might be whatever they're having taken out or biopsied, whereas post-mortems, you can take everything. So for the kidney cell atlas, what we principally have used are tumor nephrectomy samples. So this is where kidneys are taken out because someone has a tumor, or these organ donors where their kidneys actually have been taken initially to be transplanted, and for a variety of reasons. So for example, there may be a suspicion of malignancy in the donor, they can't be used. And so rather than applying bulk genomics, which, as I've said, if you have a kidney core, you're able to see which genes are up and down regulated in different conditions, but it doesn't give you any cellular granularity. With single cell genomics, and what we've principally done is to generate single cell suspensions and perform RNA sequencing on single cells, then you're able to, in an unbiased way, cluster different cell types according to similarities and differences in gene expression. And the two ways of doing that, so I should say, first of all, that you can either do that with cells or you can do that with nuclei. The advantage of nuclei is that you don't have to disaggregate the tissue, and so it gives you a much better reflection of what cells are in there. When you generate single cell suspensions, you inevitably lose some cell types. And that's particularly true in the kidney. So for example, cell types that are hard to get out because they're very adherent, like those podocytes that I mentioned. Okay, so then once you've got your either single cells or single nuclei, there are two options. So you can either perform plate-based methods, so where you put a single cell in each well and then you profile the transcriptome. These tend to be overall a bit more laborious, expensive, and low throughput. Alternatively, you can use a droplet-based platform, and this is predominantly what we've done, where single cells are combined with barcoded beads that have a unique oligonucleotide on them. And then you are able to force them into an oil droplet such that each cell can be identified by the associated barcode. And so in our version 1 of the Kidney Cell Atlas, we profiled human kidneys across lifespan. So we had access to some fetal human kidneys from the Human Developmental Biology resource in Newcastle in the UK. And you can see we had 6 of these between 7 and 16 weeks post-conception. And then we had some neonatal, so some infant kidneys. So these were children that had tumours and were having their kidneys removed because of that. We had some teenage kidneys from an organ donor that couldn't be used for a technical reason. And then we had a variety of adult kidneys. You can see most of, from older adults than younger adults. And these were a combination of tumour nephrectomy and the organ donors. And we performed 10x genomics single-cell RNA sequencing. Our QC pipeline was something pretty standard. You know, one really important thing, or really prominent thing, especially we've got better at this, but in making this first atlas, was that we had a lot of ambient RNA, and so particularly from proximal tubular cells that very easily die because they're high metabolic rate, and so we use SUPEX to remove those. We also, in terms of the mitochondrial gene content, whereas normally perhaps you'd have a cutoff of say 10 or 20%. Actually, for proximal tubular cells, again for the reason that I said, that they're really metabolically active and need energy, and so a lot of the kidney data that's become available from mouse and human has shown that you have to use a higher cutoff, so even 40 or 50%. And then, so ultimately you can see what we're able to generate were a lot of data on proximal tubular cells, some vasculature, non-proximal tubular parenchyma, and then some immune cells. The fetal kidney data, is just a lot cleaner and a lot better, a lot less dead cells, and a lower mitochondrial gene count in all. So post-QC, in the end, for the fetal kidney cell atlas, we generated data on 27,000 cells, and that included, in green here, the clusters are the developing nephron. So kidneys develop when the ureteric bud invades the cap mesenchyme, and that interaction results in the mesenchyme transforming to an epithelium, so to make the renal vesicle, which makes the S-shaped body, and ultimately the fetal nephron that contains all of the components I told you about in the adult kidney. So podocytes, proximal tubular cells, the loop of Henle, connecting nephron, and then collecting duct. And so you can see we were able to find those components. So from the cap mesenchyme out to these terminally differentiated cells, the podocytes, proximal tubular cells, pelvic epithelium, etc. We could also find some vascular endothelial cells, a whole lot of stromal cells, and then a large number of immune cells, and probably more than we thought we would find. And I told you that it's known that macrophages are prenatally seeded, and we could find a lot of macrophages, but surprisingly, we could also find other immune cells, including adaptive immune cells like B cells, T cells, mast cells, NK cells. And because we had samples from different developmental time points, it meant that we could sort of plot them through time. So particularly for these immune cells, we could ask, well, when do they start appearing in the kidney? So you can see that macrophages occur very early on, and then these later time points, we begin to see the adaptive immune cells appearing. And then in the adult kidney, we generated data in the end on 40,000 cells. So relative to the number of samples we had, remember it was sort of 6 versus 14. It wasn't as much, but that's, I showed you, there's a lot more cellular debris or data that we had to discount at QC. But again, we were able in green to see elements of the nephron. And you can see this is really dominated by the proximal tubular cells, this huge gloop here. There were some stromal cells, the vascular endothelial cells, including the glomerular endothelium, that specialized endothelium. We did have a few podocytes, but you can see not so many. And then again, a landscape of immune cells that not only included mononuclear phagocytes, so macrophages and dendritic cells, but also actually lots of T cells, NK cells, and some B cells. And what we did with this data was actually we used depth data from bulk RNA-seq and to refer back to our biopsies so that we could project into pseudospace where these were taken from and hence where the cells came from. And so we could show that sort of as a sense check, if you look in the cortex, there's lots of proximal tubules, which is what you'd expect, lots of the podocytes, which then go away as you go deeper. But for the immune cells, we could then plot, let's say, for example, up in the cortex, we can find the B cells and mononuclear phagocytes. Down here, more of the T cells and NK cells. So one thing my lab is particularly interested in is in these mononuclear phagocytes. And I said, we've got the division of macrophages and dendritic cells based on mouse models. So when we reclustered these, we could find 4 different subsets of mononuclear phagocytes, which we labeled MMP-A to D. And based on canonical markers and our subsequent work, we annotated these MMP-A and B as being monocyte-derived macrophages. And this little MMP-D population, a tissue-resident macrophage that we think could potentially be the human version of a yolk sac-seeded macrophage. MMP-C express lots of DC markers, including a very high level of MHC class 2. And in the fetal kidney, we reclustered or did fine annotation of the myeloid compartment and could find a number of different macrophage populations, one of which was more inflamed than the other, but also DCs and some monocytes, mast cells, and neutrophils. But because we had that data, we could actually use it to address the question really for the first time in humans. Well, does it look as if any of these macrophage populations that are in the human kidney were prenatally seeded? And we did that by saying, well, are any of the ones in the mature kidney transcriptionally similar to the macrophages in the fetal kidney? And what we found, there was only one subset that was really, you can see here was MMP-D. And so which is why we annotated it as being tissue resident, whereas you can see MMP-A and B look most like fetal monocytes, so consistent with our conclusion that they're monocyte derived. And of course, what we can do when we have this transcriptional information is begin to infer functionality. And we did this by looking at gene set enrichment within the different subsets. And you can see in the mature kidney that these different MMP subsets are, transcriptionally, they look functionally very different. So MMP-A has got marked expression of defense response to bacterium genes, whereas these tissue-resident MACs, MMP-D, do not. And I meant to point out to you that if we look at the markers, one of the markers we'd used previously for FACS studies was CD14, and actually that's expressed on both of these. So we wouldn't have been able to distinguish that, but our data using the single cell transcriptome showed us that these, CD14 positive cell subsets are completely different. And we went on to validate this functionally. So we asked, well, this seems to suggest that MMP-A would be phagocytic and MMP-D maybe not. And so we took single cell suspensions from kidneys and fed fluorescently labeled uropathogenic E. coli, so a bacteria, and asked, well, how do they take it up? What's the percentage phagocytosis? So you can see MMP-As very avidly take this up, MMP-Ds really not so much. And then the other thing that we were able to ask, so I guess in immunology, we focus very much on the immune functionality of immune cells and don't think so much about whether some of the structural cells, so including the epithelial cells within organs, may have immune functionality. But with that data set, we were able to ask that. And one of the remarkable things that we found was that if we looked at the different nephron components, so going from I'll just remind you, going sort of from here, the glomerular endothelium, the podocytes, through the proximal tubule, down to the connecting nephron, into the pelvic epithelium. So if we look from proximal to distal in the kidney, we could find that actually the pelvic epithelium had very high expression of a number of innate response genes, including antimicrobial peptides. So these are molecules that in and of themselves have a defensive capability. And we listed some of them here, and you can see marked expression. And again, because we had fetal data, we could compare that to the adult data. And you can see that this acquisition of innate immune capability really only happens in the adult kidney, or in the mature kidney, suggesting that there's potentially a cue postnatally that might trigger this, and one could hypothesize that this is potentially during microbial colonization postnatally. And then the other thing we asked, or were able to address, was the question of how the epithelium, the structural cells in the kidney, might instruct the immune cells in the kidney. And we did this using cellphone DB, which is a publicly available tool that Sarah's lab has generated, where they've curated a whole lot of receptor ligand interactions based on the literature. So you can ask, based on the expression of receptors and ligands, what would you predict the interactions may be between cell types? And one question we were particularly interested in, based on some work that we'd published previously, before we began looking at a single cell level, was that we'd done bulk RNA sequencing of cortex and medulla of human kidneys, and found that you can see a whole range of chemokines. So these are molecules that guide the movement of immune cells, that some chemokines are very highly expressed in the cortex and some are very highly expressed in the medulla. So we asked, well, in our epithelial cells, so shown here in green from podocyte proximally to the pelvic epithelium distally, what are the chemokines that are expressed, and what are the receptors that different immune cells are expressing that might allow them to respond to it? And you can see one thing that really caught our eye here was the pelvic epithelium expressed lots of different neutrophil recruiting chemokines, predicting that it would have the capacity to draw them in. And that would make sense, because neutrophils are the major circulating phagocytic cell. And we went on to validate this using confocal microscopy. So you can see, so this is the edge of the pelvic epithelium, and this is the pelvic space where urine would collect. We can see marked expression of CXCL8, so the major neutrophil recruiting chemokine in humans, and also expression of one of these antimicrobial peptides, lipocalin-2. And we went back to a mouse model and said, well, if we give an infection, if we do a kidney infection model, where do we see the neutrophils? So here marked by this lysem, the green cells, where do they gather? And you can see, again, so this is the urinary space, so in the pelvis. So it's exactly the same place where in the human we see the neutrophil recruiting chemokines that neutrophils are avidly drawn to set up a really specialized defense zone, exactly where you need it as the bacteria are coming up. So I hope I've shown you that kind of version 1 of the Human Kidney Cell Atlas identified a surprisingly heterogeneous immune cell landscape in the human kidney, and this was in homeostasis, so not in any disease. So saying all those cells there are poised to act, and we were able to predict some of their functionality based on their transcriptional profile and go on to validate that in the case I showed you of the two of the mononuclear phagocytes populations. It was notable that these cells were, many of these cells were also present prenatally, suggesting that some of them, the fetus is completely sterile. So you don't necessarily need an immune system to defend you against pathogens. So suggesting that some of these may have other roles in homeostasis. And then we also showed that the epithelial compartment actually has surprising immune functionality. So particularly I showed you the data on antimicrobial peptides and innate immune genes being expressed in the pelvic epithelium. And so overall, this data set gives us an important context or baseline to go on to understand, hopefully in subsequent studies, how that goes wrong in different kidney diseases. So for the final sort of 10 or so minutes, I want to tell you about how perhaps we could think about using the kidney cell atlas in biomedicine, and some of these things will be obvious. So one thing is to try and take our current attempts at understanding disease, so our application of biopsies and pathology, to the next level. So I mentioned, this is sort of standard clinical care. So light microscopy, immunofluorescence to look for antibodies, etc. And we sometimes, if you've got enough tissue, do EM. In some contexts, much more limited, we do it's actually a microarray based assessment that may allow us to better diagnose, for example, rejection in transplantation. But some of these signatures, so for example, the signature that's associated with antibody-mediated rejection, one of the components of it that has been focused on is that we see activation of endothelial cell transcripts. Well, there's a couple of papers that have now done RNA-seq on kidney transplant biopsies with rejection. This was the first one from Ben Humphrey's lab. So this was a single patient with mixed rejection. And they were able to identify a number of different clusters. And in fact, if you were just looking at this, you might think all of this was related to pathology, all of these immune cells. But in fact, our version 1 kidney atlas tells us immune cells doesn't equal pathology. So you have to take it in that context. But what I wanted to show you was, previous attempts at using molecular pathology in kidney transplantation have said, well, there are these endothelial transcripts that we see upregulated in antibody-mediated rejection, and there are T-cell mediated transcripts. Well, if you look at these so-called endothelial transcripts at a single cell level, actually a whole bunch of them are not endothelial at all. So, proximal tubular cells are what's responding in this. There are some that are endothelial, but mast cells are responding, myofibroblasts are responding. So it kind of completely alters our ability to understand the molecular context of pathology. The other type, or the other thing that we can do is we can begin to map genome-wide association study data onto cell-specific expression data. So for example, in chronic kidney disease and kidney function, there have been a number of genome-wide association studies that have identified specific polymorphisms in different genes that are associated with susceptibility to either a worse kidney function or to chronic kidney disease. So what we can now do is say, well, what cells within the kidney are these GWAS hits expressed in? And again, the value of this is that it begins to take us to focus on specific cells at which molecular pathology may be focused to bring about disease. The other way we can use this data to improve our insights into disease pathogenesis is in a way in which we start with a hypothesis. So for example, I've mentioned a few times that antibodies can cause disease, and they can cause glomerulonephritis, or they can cause antibody-mediated rejection. So we know in the case of IgG antibodies that one of the ways in which they activate immune cells is by binding to surface IgG receptors. And there's a whole family of these, and they're called Fc gamma receptors, the gamma referring to the fact they bind to IgG. So using our version 1 kidney cell atlas data, we can actually ask specifically which are the cell types that are going to respond to antibodies. So which are expressing the Fc gamma receptors, and there are also different types of these. So one of these is inhibitory and the others are activating. So we can pick up, perhaps surprisingly, NK cells that are resident in the kidney are poised there waiting to respond to these antibodies. NKT is also expressing monocyte-derived macrophages, major expressors of Fc gamma R3A, others expressing Fc gamma R2A, and so I should say coming online therapeutically are antibodies that specifically block these interactions. So again, we can begin to move towards precision medicine. And then the final thing which I wanted to highlight is the utility of our developing kidney cell atlas. So, remember I told you that we were able to map everything from the cap mesenchyme, where it all starts, through to terminally differentiated cell types like podocytes or proximal tubular cells, pelvic epithelium. And we can set this out in pseudo time and actually map cell state trajectories from, say, the cap mesenchyme right through to the podocyte, and then plot out the transcription factors that lie along that cell state trajectory. And this information is extremely valuable, because if you wanted in the laboratory to make a terminally differentiated podocyte, you know exactly the transcription factors that you need to turn on to get to that place. So it more or less gives us a specific recipe of how we could make every single terminally differentiated cell type in the kidney, with the potential to rebuild organs either from scaffolds or de novo. Hopefully I've shown you how the Human Kidney Cell Atlas has revealed really quite unexpected complexity in the resident immune cell landscape in normal kidneys, showing us that perhaps our naive assumption that the presence of immune cells, particularly adaptive immune cells, were always equated with pathology is not true, and to give us something that acts as a reference for our subsequent single cell studies in disease. It also allows us to take both nephro and transplant pathology to the next level. So we go from current molecular pathology, where we might see sets of genes upregulated and we don't know which cell type is expressing them, to cell-specific granularity. Similarly, in terms of genetic associations with disease, we can now relate those to cell type specific expression in the kidney, and that sometimes gives us surprises. And then we can also use it when we have hypotheses, such as, we know that Fc gamma receptors are involved in antibody-mediated pathology, so which cell types are expressing them. And then finally, it provides a transcriptional map of how cells change during kidney development to produce terminally differentiated cell types that can be used in regenerative medicine. So to the acknowledgement, so it's obviously quite a large team was involved in this. So in my lab, the two people that drove the data analysis were Ben Stewart, who's just coming to the end of his PhD, and John Ferdinand, a senior postdoc in the lab. And then there were a number of individuals that helped with processing the kidneys. You can imagine some of those come at all times of day, the transplant donor ones. So particularly Kevin Loudon, and also previously Alex Riding and Andrei Suchanek. And then my collaborators, so particularly Sarah Teichmann at the Sanger, who's been incredibly supportive and a really dynamic force to generate a number of the organ cell atlases. And then also, I particularly would like to acknowledge Sam Bajhati, who's a pediatrician who accessed those pediatric kidneys, and Maz Hanifa, who is based in Newcastle and was involved in accessing the samples from the Human Developmental Bioresource. And I would also like to thank the patients who undergo nephrectomies, and yet in that traumatic time are willing to give us samples, and also to the organ donors and their families who, you know, in a time of difficulty, maybe want to see something good coming out of what isn't a great situation. And then to the various funding bodies. And finally, our data is available for all to access. So first of all, if you have informatic capability and want to access the raw data, that's available via the HCA data coordination platform. And we've made all of our data so far, all of our experiments so far are available. If you just want to have a quick look at which cell types are expressing your gene of interest, then you can do that at kidneycellatlas.org, where I mean, it's pretty easy to navigate, but we have all of the data from both fetal and mature kidney. And with that, I'm very happy to take any questions. Thank you.
Thank you very much, Dr. Mena, for your wonderful talk. We have some questions from the attendees. The first question from an anonymous attendee. Thank you very much for this great talk. The kidney is such a complex organ with such different specialized cells. How do you suggest to start profiling if you were to design a first project?
Well, I think you know, one thing is it depends what you're interested in. You know, when we were going for version 1 of the Cell Atlas, we knew we wanted to have some immune cells in there because that's my lab's interest, but those cells are rarer. And so what we did was after we generated the single cell suspensions, we used a Percol gradient, which actually gets rid of the dead cells and removes some of the epithelial cells, so enriches for the immune cells. And then in the fetal kidneys, we actually sorted some CD45 positive cells. So it depends what you're interested in. If you're interested in podocytes, you know, then those are very rare cells. And so you'd have to think about how you enrich for those, whether that might be by sorting them or by isolating glomeruli and then getting them.
Thank you. Another talk from another anonymous participant. Great talk. I have family members with recurrent urinary tract infection. It gives extra hope to see how much evolution in the basic research. Have you seen any polymorphism that might play a role in the cellular response to kidney infection?
Yeah. So that's a great question, and whoever asked that. I mean, one thing that's continues to amaze me working in this area is that although urinary tract infections are phenomenally common and they can actually cause chronic kidney disease and even end-stage renal failure, the amount of research into urinary tract infections is actually relatively small. Now, one might speculate that's because, you know, it's 50% of women that get urinary tract infections, not men, but that's perhaps another issue. So there are currently, there's only one genome-wide association study that's been published in urinary tract infection. And that was done using the 23andMe data. And that only identified two polymorphisms, or two polymorphisms that are associated, and neither of which were highly expressed in our data sets, one of which is in a non-coding region. And I think one of the problems with that data set, it put together all urinary tract infections, so both bladder and kidney. And I think that it will be important to get bigger data sets that have collections of patients with kidney infection and those bladder infection, because I think there are separate susceptibilities. We're actually trying to do that very thing using the UK biobank. So this is a biobank of 500,000 people in the UK that have donated samples and said that, and basically they've had SNP genotyping done. And then they also have phenotypic information like infection.
Thank you. We have another question from Tiago Lubiana. Do you think there are still immune cell types to be found in the kidney? How much diversity might be still hidden?
So I think that there will be. there's a whole question of cell types versus cell states, and there may be some cell states are associated with disease. So in the brain, you have these disease-associated microglia, and I think there may be states in the kidney too. I think what we'll, even in our data set, for example, I showed you the number of macrophages that we had was relatively small. And I believe in that tissue resident subset, there may be more heterogeneity. So again, in mouse, some macrophages have been found to be associated, for example, with blood vessels or nerves. So it's going to need us to get better at getting them out alive and having different enrichment strategies.
Thank you. Another anonymous participant, great talk. How far are we from applying these results or ideas in the clinical practice, in pathology, let's say?
Yeah. I think in reality, for the big problem, right, with single cell technologies at the moment is that they're expensive, and the reality is we struggle to implement them in an equitable way, even in research. So if we're talking about trying to apply them widely clinically, certainly in the UK, we have a public healthcare system that's centrally funded. It simply couldn't stretch to this. But I think the first step is for researchers like myself to begin to utilize samples from biopsies, from clinical biopsies of patients with kidney diseases. And we've started to do that. The challenge is when you take a piece of kidney from a person, the amount you get is small compared to the stuff when we have a whole kidney, we can take whatever. So that's really, that getting enough cells to be able to take them, through to get useful information at the end is what we're working on. But I think there are examples of that. So for example, there was a very nice data set in lupus nephritis that was published in Nature Immunology a year or so ago. So, I think we will get there. So the first steps will be for us as clinicians, scientists, and as translational researchers to do it. And then with time, hopefully things will become cheap enough to be able to actually use it in the clinic.
Yes, of course. Another question is, have you estimated the single cell expression profile of lung non-coding RNAs in the kidney, your group, or have you seen another group?
I didn't quite get that. What was that?
Have you estimated the single cell expression profile of lung non-coding RNAs in the kidney?
No. No, that's not something that we've looked at.
Another question is, do you think it will be possible to trace back some of the issues of recurrent infection to host pathogen interactions at the single type level?
Well, there's obviously been a lot of interest in relating certainly viral transcripts, right, to the cell in which they're found in COVID-19. So for to try and look for viral related transcripts. So I think that would be possible for some viral infections in the kidney to associate them with specific cells. And there is a virus called BK virus that infects the kidney very commonly, and I think that would be possible. I think for bacteria, that's harder, although, E. coli, which is the commonest cause of bacterial infection in kidneys, it does get into epithelial cells. But at the moment, I know that there are efforts to try and do it, but I'm not aware of any efforts that have managed to get bacterial genome or transcripts out of a cell and the cellular transcripts from the same cell.
Thank you. That's it. Thank you very much, Dr. Mena, for your wonderful talk. It was a pleasure to hear you here in this event.
Thank you very much for inviting me. So it's a pleasure to be here, and I wish you well with the rest of your workshop. Bye now.
Thank you. Bye-bye. Now, guys, I'd like to invite Patricia and Enrique Lemus just for some final words for this first week. Are you there?
Yes.
Hi, Enrique.
Hi.
Hi, Patricia.
Hi, Vinicius and Enrique.
So we are the last day of our first week.
Yes.
It was a long week, guys. Thank you very much for your participation. It was a pleasure to see a lot of people, a lot of involvement. Unfortunately, for the second week, we had a small number of space to select, so we are only working with 30 people, but I'm sure that will be a wonderful second week. Patricia would like to say some words for the people who attended the event for only this first week.
Yes, I would like always to remind everybody that this is a follow-up on the first event that we had. So there was a lot of contribution from the colleagues during that event for us to come up with these ideas, and the workshop for training, which is a first opportunity for Latin America to get involved with the Human Cell Atlas until we have our infrastructure ready and our own data sets ready to be analyzed. And the idea was to join first 3 countries, because this is a prototype of the way we would like to work. But we do have a lot of people from Peru and Argentina also interested in joining. So hopefully the next one will have more groups, more countries with local trainers also in order to engage the local communities more and more. And also hopefully next year we're gonna have people from Latin America showing our own results, either of data analysis or data that we have generated during this next month. But thank you very much for being here. We looked for speakers very carefully for people who are really experts and great researchers that would be available also to talk to you guys as a follow-up. So please reach out if you're interested in what they presented to us. They are very eager to help Latin America join the Human Cell Atlas.
And before to pass the word to Enrique, we will be sending you guys a survey, so you can give your thoughts on how it was, the event. As Patricia mentioned, this was a pilot one. The idea is to repeat it in other regions, not only Latin America. And also the idea is to start to build capacity in this field locally with small events, small courses that each country can help themselves, not only together, everybody together, but and also to put people together, to know ourselves. The idea is to start to work on this in Latin America, to start novel collaborations in this field. And Enrique would like to say some words.
Yes, first of all, thank you for all of you for your attendance to this week. It has been a long week, but also I think that has been a quite interesting overview and introduction to the field. And it's my impression that it was a balanced view between the clinical applications, the biology and the computational biology aspects, the data analysis, and even those who won't be attending next week, the second week, my wish, my impression also is that it could result also beneficial for all of you to get a glimpse, to get a grasp, an introduction to this new field, and several new ideas hopefully will develop from this. So don't lose touch. Please stay in touch, and perhaps we are thinking that those that will be trained during the second week will become like the next generation of trainers. So we will be spreading this further. So keep in touch, and thank you for your attendance to this workshop.